Try to be on time. Second speaker, Angela Huang, she is not able to make it live, but she has sent us her video recordings that we will be playing. And then once we do this, then towards the end, we will have some time and there will be more questions for you. But it's yeah, so everyone please ask questions. There's no such thing as a dumb question. Everyone please ask questions. You can type them in the Q&A. You can chat as well, and yeah, so on and so forth. So we will soon be starting. So it's still, we have a minute or so. And yeah, so Lucy, you get ready. You'll get going in a minute.
Okay. So I should share my screen now?
Yeah, I think I will stop sharing my screen. Now you can share your screen.
Okay. Okay.
Yeah. So so we have the first speaker, Yikin Lucy Liu. She is PhD student at the University of Engineering, Science and Technology, China, and she will be talking about medial prefrontal activity at encoding determines the recognition advantage of threatening faces after 1.5 years. So Lucy, it's all yours.
Okay, hi everyone, I'm Lucy, and it's a great pleasure to present my PhD work here. So today's topic is about the neural basis, especially during encoding of the recognition of threatening faces with a very long retention interval of 1.5 years. So faces are arguably one of the most important stimuli for social interactions. It contains a variety of information, including age, gender, and emotional expression. More importantly, it may transmit survival-related information. For example, when we see threatening faces, we might want to avoid it, and even in the very long future. So detecting and remembering threatening faces is extremely vital. Experimental studies have provided evidence on the threatening advantage from working memory, short memory to long-term memory, while the working memory and short-term memory showed consistent results on threatening advantage. But long-term memory advantage of threatening faces remains uninvestigated, especially with a very long retention interval. So here in our study, we address this question by designing a behavioral experiment and fMRI scanning. So basically, in the first stage, the participants were underwent fMRI scanning, during which they had to judge the gender of the faces. There were 5 emotional categories of the faces, including angry, fearful, happy, neutral, and sad, and the gender were balanced. And after like 20 minutes, they were asked to finish an immediate recognition test in which they were required to determine if the presented face was an old one or a new one. Most importantly, we are interested in the long-term, in the delayed test after 1.5 years. So after a very long retention interval, the same group of participants were called back again and to complete another surprise recognition task and with another set of new faces intermixed. And to analyze the data, we firstly look at the behavior data by using univariate ANOVA analysis. And moreover, we were interested in the multivariate pattern of the behavioral response. We specifically use PCA to decode the participants' behavior response. What's more, we, based on the behavior results, we look at the neural basis during the encoding that might contribute to the later recognition of the emotional phases. So the result, first, in general, we can see the participants successfully discriminated the old phases from new phases by the A' index. In two delayed and immediate tests that both showed a very higher level than the higher A' than the chance level. And the paired t-test suggested that the discrimination ability decreased after the 1.5 years. What we are more interested in is the emotional expression effect on the face recognition. So in the first step, we look at the main effect on the emotional expression in delayed test, and ANOVA showed a main effect. The post-T tests suggested there is the angry and fearful faces showed a higher or better recognition than the happy and neutral faces. However, the sad is just in between of the two, of angry, fearful, and happy and neutral. So these results indicated a long-term face recognition advantage of the threatening expressions on the face recognition. The comparison with the immediate tests further suggested that the recognition advantage of threatening faces was driven by the decreased recognition of the non-threatening expressions, including happy, neutral, and sad. Okay. So based on the univariate results, we can see a very good or clear pattern that the threatening faces were better remembered. However, given that the univariate analysis only used the averaging data, but ignored the individual response patterns of the whole trials. So to complement this, we further investigate the response patterns behaviorally on the confidence ratings of the faces. So we can see from the left figure, there were great individual differences on the response patterns of the 5 emotional categories. So to try to dig out the hidden pattern of the responses, we use PCA to do that, plotting the confidence rating of the trials, which is served as the features of the data-driven approach. And then we expected to see a clear separation between the 5 emotional categories. And yes, we found a similar or the same patterns that we found from the univariate analysis. And we can see from the left figure, the scatter plot, the 5 colors indicate 5 different emotion categories, and the angry, especially angry and fearful, cluster more closer, and happy neutral is clustered closer, while sad was just in between. Also from the distribution, we can see the same pattern. And also the first component on the x-axis just represent the most discriminative patterns of the emotional expression. So the both univariate and multivariate results suggested a same direction of the recognition on the threatening versus non-threatening expressions. But we wonder if all participants or each participant share the same discrimination ability on the emotional expression. So we did pattern similarity analysis on the behavior data. So what we did is to firstly extract the PC1 score from the PC analysis. This PC1 score can be used as a response template, which represents the most discriminative pattern of the 5 emotional expressions. Then we correlated the PC1 template with each participant's response patterns so that we can determine if the participant shared the similar pattern as the very first principal component. And then based on the significance, we can split the sample into 2 subgroups. And the one group with the significant correlation or the similarity can be viewed as discriminators who can discriminate different emotional expressions, but the other group would be the non-discriminators. So behaviorally, we also confirmed our predictions. So based on the 2 groups, the discriminators shared the same differentiated recognition confidence on the 3 groups of emotional categories, but not in the non-discriminatory groups. So within the 2 groups, we can did fMRI analysis to test the neural basis underlying the discriminative emotional face recognition, especially on the face expression. And the first level contrast is just generated or obtained from the behavior results, which is the angry, fearful minus happy and neutral, because these 2 clusters are the most different. And then we we did a group level with comparing the discriminators with non-discriminators, and the whole brain ANOVA models just detect a significant cluster in the medial prefrontal cortex. And in this region, it suggested that in the discriminator groups, they have had a decreased activation towards threatening versus non-threatening faces. And also, we extracted the parameter estimates from this cluster. We can see the pattern in this. So these results suggested that decreased VMPFC activity to threatening faces during coding might contribute to the maintenance of long-term memory for threatening faces. So to conclude, we firstly confirmed a long-term memory advantage of threatening faces versus non-threatening faces after a long-term interval of 1.5 years. And neurally, we found decreased prefrontal reactivity to threatening faces during coding might contribute to the maintenance of long-term memory for threatening faces. And these findings have implications for understanding the emotional memory model and also the memory dysfunction of affective disorders. Thank you for your attention. That's my talk.
Thank you. Thanks a lot, Lucy. It was great, and you timed it very well as well, so you're on time. So we have actually one question coming from the audience. So, so Jin Ki, so I can ask probably on behalf. So the question is this: that have other principal components been explored or explained? So because you have looked only at initial few PCs, PC1 and PC2.
Yes, okay, this is a good question. I think we did other PC has been, yes, we also tested the gender of the faces, which is the main features involved in the faces, and we found that the gender could be the second principal component, but explained less variance than the first one, which is the emotional expressions.
So just to follow up on that, so what is the distribution? What was the demographics of your, so what were the age ranges and the gender and of the participants?
Participants, okay. Our participants are mainly undergraduate or graduate students, so they are young, healthy participants, and I think the gender is just half and half based on this. So, what's your question?
Is there any other question? I have a quick one. So what you see is you have VMPFC, which is quite so you head in the right VMPFC. So any
Yeah.
reason why you should have this much lateral, on one side? I would have imagined it maybe the left VMPFC. And because it's about emotions, I thought there may be amygdala and insula involved as well, subcortical regions. So we don't see them. Did you see any of those regions?
Yes, on the brain level, whole brain level, we only detect the right OFC region. But we also hypothesized that amygdala might involve in this emotional process, and so we did a RI analysis, but also didn't find a significant cluster.
Okay, maybe we could continue it later on. Thanks a lot, Lucy. Thanks a lot. It was great. Well done. I very enjoyed your talk. Okay, so let's move on to the next talk, which is from Angela Huang. Angela is a PhD student at University College London, and her talk title is Reward Specific Activation in Prefrontal Cortex During a Gambling Task. Angela had some problems with her internet, so she can't do the live session, but she has sent us a pre-recorded video that we will be playing. Unfortunately, of course, she can't. I don't see her here, so it means she can't give a live answers to the questions. But at least we have her talk. So yeah, we will start with Angela now. Thank you.
Hello, my name is Angela, and I'm a third-year psychology undergraduate at University College London. I'm very delighted to participate in Neuromatch conference, but my internet router has not been working properly recently, so I couldn't join the live Zoom session. Please excuse me for this pre-recorded presentation. Okay, so today I'm going to talk about reward-specific activation in prefrontal cortex during a gambling task. First, I'm going to start the presentation by introducing several past studies on prefrontal area, such as ventromedial and dorsolateral prefrontal cortex. Then I'll be talking about the human connectome dataset and method of the present study, followed by results and conclusion for this presentation. An important function of the prefrontal cortex is the control of goal-directed behavior. For example, we have to go through decision-making procedure to choose the most optimal option for our goal in given situation. In that decision-making process, we of course compare and choose a rewarding options than other less rewarding options. This decision-making process then leads to behavior and help us reach the goal in everyday lives. Among several prefrontal areas, today, I would like to focus on dorsolateral prefrontal cortex and ventromedial prefrontal cortex today. The reason that we investigated these two regions will be explained in the method part. First, let's talk about the dorsolateral prefrontal cortex. The dorsolateral prefrontal cortex is essential for sensory and motor system structures and is also involved in cognitive functions such as attention, working memory. Initially, many functional neuroimaging studies in humans have supported a role of dorsolateral prefrontal cortex, I'll just call it DL cortex, in working memory. In the Charles research in 1998, subjects with right DL cortex lesions were impaired on the delay tasks, but not on the gambling task. During the delay task, subjects were first asked to pay attention to the 2 open cards from 4 cards before they disappeared from the screen. When the cards are disappeared for 1 second and reappeared, participants had to choose 2 cards that they were told to pay attention. When they chose the correct cards, they got $100 on the computer screen, and if they make a mistake, the $100 was deducted. For gambling tasks, participants were asked to win as much money as possible by choosing cards from 4 decks of cards. 2 of the card decks had high incentive, but with even higher penalty that led to negative balance, and other 2 card decks had low incentive with lower penalty, which leads to positive balance. Participants had to learn this pattern by themselves. So, as a result, in this study, subjects with right DL lesions were impaired on the delay tasks, but not on the gambling task. Yet, because subjects with DL lesions also performed at low level during gambling tasks, it was concluded that decision-making seems to be influenced by the intactness or impairment of working memory. Now, let's move on to the other prefrontal area of interest, ventromedial prefrontal cortex. It has been studied that ventromedial prefrontal cortex damage often leads to unstable control of emotion. However, many studies have also investigated ventromedial prefrontal cortex and its role in decision-making process. First, an extensive amount of studies have reported that humans with damaged ventromedial prefrontal cortex showed severe impairments in decision-making. In fact, a significant amount of studies have reported that the irrational choice of ultimatum game offers has been correlated with feelings of anger, skin conductance responses, and negative feelings represented by activation in insula. Ultimatum game is a notable experimental paradigm used for economic decision-making. During the experiment, 2 players have to split a sum of money. One player offers a portion of the money to the second player, then that second player can accept the offer, which leads to splitting money, or reject the offer, which both of them gets nothing. Rational choice would be responder accepting any offer regardless of the split ratio. So using this paradigm, Konix and Trano showed that participants with adult onset bilateral ventromedial prefrontal cortex damage were more likely to reject the offer than the control group. This fMRI study indicated that emotional response is critically involved in the responder's decision-making process. Okay, so among reward-related studies of prefrontal cortex, we have noticed that there isn't many investigations that focused on the reward-related activation, emotion, and gambling task. Therefore, during the NeuroMatch Academy project, we came up with this research question. Do activation differences in prefrontal cortex correlate to differences in each condition of the gambling task? In human connectome dataset, we used several prefrontal regions were combined with other areas in terms of parcellation. Therefore, for the research question, we specified as prefrontal cortex instead of ventromedial prefrontal cortex and dorsolateral prefrontal cortex. We have predicted that there will be a significant difference in prefrontal cortex between win and loss condition. Unlike previous studies, which involved subjects with frontal damage or lesion, our study, which used HCP dataset, involved 77 healthy participants comprised of 58 female and 19 male participants. During the task-based fMRI HCP data collection, Delgado's experimental paradigm for gambling task was used. The paradigm involved a series of 32 trials in total, divided into 4 runs of 8 trials each. Each trial starts with a choice-making period. Participant was presented with card projected onto screen. The unknown numbers written on the card ranged from 1 to 9. When the number wasn't shown, participant was asked to anticipate the number written on the card. Participant had 2.5 seconds to answer whether the number is higher or lower than 5. If the response is not made, then the trial was excluded from the analysis. Then participants were instructed to press the left or right button of a response unit to select the answer. After they pressed the button, the number written on the card was displayed. If a green arrow pointing upward is shown, it's indicated that it was a correct guess, which gives a reward of a $1. On the other hand, if the red arrow pointing downward is presented, that indicated that it was an incorrect guess and the participant got a penalty of 50 cents. If the number displayed was exactly 5, then participant neither won nor lost the money. However, whether each trial is going to be reward, punishment, neutral event was already predetermined and it was unknown to the participant. For example, 2 blocks were mostly rewarded and the other 2 blocks was mostly punishment event. Therefore, this predetermined events lead the participants to experience negative feelings during the mostly punishment blocks and positive feelings during the mostly rewarded blocks. So for the fMRI data analysis, we compared the activity in the parcels of the differences between the win and loss conditions for significance with a t-test for unpaired samples. First, as you can see from this figure, there was a huge difference between win and loss condition. To explain this a bit further, it is a comparison between significant win neutral and loss neutral condition. Therefore, these regions were all the regions that had significant difference in activation. Now, this 3D image of the brain is showing the significant regions which have been corrected for multiple comparisons. As you can see from the image of the brain that is showing both hemisphere on the very right side, ventromedial cortex and ventrolateral area show a very significant brain activation. Now, when we conducted a correlation cluster analysis, functional connectivity plot shows that the frontal parietal area is significantly correlated with both win and loss conditions. In conclusion, a significant activation in prefrontal area indicates that prefrontal cortex is involved in monetary reward processing, and this was in line with the past literature. Also, different activation in each predetermined win and loss during the gambling task might imply dissociation in negative and positive outcome evaluation process. And lastly, it may have implications for understanding the control of emotion in ventromedial prefrontal cortex and process of pathological gambling. Okay, so that was the end of my presentation, and thank you so much for your time. I apologize again for the pre-recorded presentation due to technical problems. And finally, I would like to say thank you for people who helped my very first fMRI project during this summer. Thank you.
Okay, thank you. So it was a great talk. Actually, it was very clear, and yeah, very nice. Very well explained. So we are actually on time. So we of course we can't have live Q&A. So we then move on to the next talk, which will be given by Vidisha Bansal. And she is a research assistant at Dongmyong University, Busan, South Korea. And she will be talking about rediscovering the pathways in schizophrenia is essence of neurotransmitters and antipsychotics. So over to you, Vidisha.
Thank you. Hello everyone, my name is Vidisha Bansal and I'm a chemical engineer. And today's topic for discussion is rediscovering the pathways in schizophrenia and essence of neurotransmitters and antipsychotic. As we know, schizophrenia is the most widely recognized mental disorder. Approximately 1% of the entire population is affected through this by this mental disorder. There are many characterizations which are being discussed nowadays to diagnose schizophrenia, but the most important which are stated by World Health Organization are disorganization in thought, emotions, and behavior. More than 1.5 million studies are reported on schizophrenia, which widely includes causes, symptoms, pathways of different neurotransmitters and causes, treatment, diagnosis, and also few studies state about the post-treatment and post behavior of patients gone through schizophrenia. There are 5 major causes which are discussed on schizophrenia are genetics, neurotransmitters, environmental factors, lifestyle, and antipsychotic drugs. Now, according to the causes and the symptoms seen by the patients or diagnosed by the doctors, there are 5 types of schizophrenia: paranoid, disorganized, catatonic, undifferentiated, and residual. Now, these symptoms which a patient suffers during a schizophrenia are further divided into 3 types: positive, negative, and cognitive. These disorders are very progressive as the disease progress, and there are at times a stage come where there is no differentiation between the real and the imaginary, which actually leads to self-harm and suicidal thoughts and even suicidal activities. Now, while going through different literatures available on schizophrenia, I identified and realized that there are some questions unanswered, which include that the identification of different pathways or changes in the pathways due to dysfunctioning of neurotransmitters in the brain, how the variation of neural pathways affect the state of symptoms, that with time how they are aggressive, and how the different neurotransmitters do set some symptoms, to identify changes in the pathway due to administration of antipsychotics, that how do the antipsychotics actually hold on or suppress the symptoms and treat the patient. The dopamine hypothesis is one of the most relevant and promising hypotheses till date for understanding schizophrenia and its symptoms. Out of all the 5 receptors, D2 receptor is the most involved, and D1 is being investigated nowadays. Brain region which is mostly affected by or stated in the dopamine hypothesis is thalamus, due to which there are some severe symptoms which are related to positive. And also the antipsychotic drug studies state that only 60% of positive symptoms are reduced or suppressed during when we treat dopamine receptors through antipsychotic drugs. We realize that the relation between negative symptoms and cognitive symptoms in the dopamine hypothesis is still into consideration or needs to be explored. So we are still working on it, and there could be some connection according to that. Now, this is a systematic representation of dopamine hypothesis in schizophrenia, that when the dopamine regulation is normal, there is normal functioning, and that is healthy psychotic, that is healthy brain functioning. When there is some dysregulation, which is usually preferred, which is actually stated that the secretion of dopamine is increased in various parts of brain, which leads to dysfunctioning in the brain. So that is and causing schizophrenia symptoms. This is from our previous paper, which is in press right now. Then the second most major hypothesis is about glutamate. Glutamate hypothesis is nowadays very commonly talked because it associates with all the three types of symptoms. So all the positive, negative, and cognitive, they are being talked into this hypothesis, not just like dopamine, they're only focusing on positive. Relationship between glutamate and various genes are also being investigated in different studies. And these genes are also related to different neurotransmitters other than glutamate, like GABA and dopamine. So, and symptoms reductions are also seen that the symptoms which are there in schizophrenia, which are diagnosed in schizophrenia, do get suppressed when glutamate is treated through antipsychotic drugs. Then NMDA, that is NMDA, the receptor of glutamate. It was noticed that in the previous studies that any abnormalities in NDMA could lead to dopamine and GABA dysfunctioning, which can cause severe symptoms of schizophrenia. Then the drugs are typically differentiated into atypical and typical. Atypical are the newer drugs for schizophrenia, for treating schizophrenia. These newer drugs are more efficient because they have fewer side effects with the other drugs which you take, which a patient is given on the diagnosis of different problems like obesities, diabetes, cancer, even heart-related issues. Because with schizophrenia, these diseases are commonly appearing in patients. Then typical drugs are the older drugs which were given previously, but they had some severe side effects with now that they are not preferred much, but still they are good prescription drugs till date for the patients who suffer a severe symptoms of schizophrenia. Other drugs like antidepression and anti-infection are also given to the patient to prevent other problems during schizophrenia. Antipsychotic drugs are helpful in reducing mostly the positive symptoms. The studies on negative symptoms and cognitive symptoms are still into studies, are still into consideration. And mostly papers talk about the positive symptoms, and both the drugs act both as antagonist and agonist. So we have to focus on that which drug is acting as agonist and antagonist, so that we could actually follow the pathway after the antipsychotic drugs are given, so that we could actually say that how the drugs are working in the brain and suppressing the schizophrenia symptoms. Alternative pathways are being noticed. There are few studies which states that after the antipsychotic drugs are given, on the basis of their nature, whether they are antagonist or agonist, there are alternative pathways noticed in the brain. But to firmly stand on this, we are still in progress. This work is in progress right now. It is not yet completed. So to stand firmly on this point, we'll be needing more research and time to present this point. Work done till date are that we have identified neurotransmitters for different symptoms, identified different pathways for different neurotransmitters. We have traced some changes in the state of symptoms due to alteration in neural pathways. And we have also identified various brain regions affected during schizophrenia from different neuroimaging imagining studies. Work in progress are identifying, we have to identify the widely used recent antipsychotic drugs as antagonist and agonist, so that we could further trace the changes in the symptoms which are after consuming the antipsychotic drugs, then identify of any changes in the pathway after consuming antipsychotic drugs. The suggestions over here are invited. This work is still in progress, and we have presented it to get insightful and helpful comments and suggestions from the experts listening this presentation. And I would also like to thank my lab members and my supervisor, Dr. Indana Chatterjee, and other lab members for helping out in this work. Thank you.
Okay, that's great. Thank you, Vidisha. So, yeah, so you have done a literature survey looking at different neural pathways, identified them, different brain areas that may be implicated. So that's great. So let's see if there are any questions from the audience, people who are attending. So there seems like to be one question. So actually it is from, so I should actually say we have at back end, we have Hadi Motamidi, and he's actually the person who is making these things happen, although he's not on in the front. So he's at the back end helping us running this session. And actually he is very excited about science as well, what's happening here. So he has a question for Vidisha. So how are these pathways studied? And can we study a pathway for two or more neurotransmitters simultaneously?
That's a very interesting question, and thank you for raising this. Indeed, the pathways which are right now studying we are focusing on the literature already available, and then we are going to give a theoretical insight on if there are different pathways also available, because the entire the study is on making the pathway possible, the possibilities of having different pathways, alternative pathways. And the second part of the question, as it says, that can we study a pathway for two or more?
So that's, yeah, exactly. So the second part of the question was this, that can we study more than one neurotransmitters simultaneously?
We are actually focusing on the interaction, the inter-interaction of different neurotransmitters. Like there are studies focusing on the interaction between dopamine and GABA, especially dopamine and GABA, and serotonin and dopamine. So the studies on simultaneously focusing on two different neurotransmitters or more than two is a very intensive study, and they need a lot of path recognition of different pathways which they are involving. Like if I remember right now, one example, that there is a gene known as DARP. This gene is responsible to manage NMDA, the receptor of glutamate, and D1, the receptor of dopamine. So when we are talking about one gene, if there's any fault in one gene, then there are two receptors getting disturbed due to that. So there are different we can study different neurotransmitter synapse at one time simultaneously with help of different genes, or maybe some different pathways which are there. So it is possible. I hope that
Okay,
answers
that's
the question.
good. I think that's more than will do. Okay, so any more questions? So probably people are still thinking. So because you are the only speaker remaining live, Lucy is not. She had to go, so she has left, the first speaker, and also Angela Huang is not here. So we can probably ask a few more questions to you. And there's another one here. So what do you mean by when you talk about positive side effects? Yeah, I mean, that was an interesting term, positive side effects.
Exactly, that was an interesting term for me as well. Positive side effects mean not something good side effect. It means that the side effects which are visible or which a person actually develops in their natural behavior, like hallucination, uh, delusions, problem in speaking. So those are positive. They are mentioned as positive symptoms, and because they are visible to the doctor or the other people who are interacting with the patient who's suffering from schizophrenia. So that is the word positive side effects of different malfunctioning of different neurotransmitters.
Yeah, I mean, hallucinations are the good trips. they are, one of the examples that you gave. So there's another question coming from Bianca von Keminade. So she has a question that do common antipsychotic drugs only target one neurotransmitter, or are there drugs that target multiple neurotransmitters?
This is actually into consideration of our study. This answer to this question will be given a bit later because this is into progress. It is not yet finished, but I believe there are drugs which have different symptoms which are responsible to suppress two or at least two neurotransmitters together, because there's a great relation between dopamine and GABA. So if they are any malfunctioning in these two, then there should be a drug available out there to suppress the symptoms relating to at least these two commonly. So to answer this, I'll need some time.
Yeah, that's good. So I had one question, which is you have focused mostly on pharmacological interventions. So what about non-pharmacological interventions? So what about, for example, meditation and exercise? So they could also have a positive impact. So what do you think about that?
Exactly. The pharmaceutical, if a patient is given treatment non-pharmaceutical, they have really positive impact on the patient to reduce some symptoms in them. My favorite, we have gone through them, a couple of them. Our study will be focusing on them also as well, but a little, not much, but a little. One of my favorite and which I can remember is music therapy. This is for the people who get very violent in nature. So music therapy is given to them, and soothing music or some instrumental music therapy is given to them because it is believed if you hear such music, the serotonin receptors are activated, and due to serotonin release in the body, you get a little suppress in your violence nature. So this is one of the study we have been focusing, and we have decided to work on this.
Yeah, that's great. I think that's good. So we are towards the end of the session now. Thanks a lot, Vidisha. I think we did great to be on time. Very interesting talks. I really enjoyed the session. Thanks a lot to everyone who contributed, both panelists and also audience who asked very interesting questions as well. So thank you. Thanks a lot. And bye-bye.
Okay, so welcome back to the NeuroMarch 3 conference. We will be presenting, we have a very nice stream of presenters lined up for today's session, this particular session especially. And we will be talking about somatosensory signals, neural divergence, and we will be also talking about interhemispheric communication during haptic self-perception. Let us start up with our first talk of today's session. The talk is by Ezgi Eriken. She is postdoctoral researcher at Justus Liebig University, Gießen. She works in the lab of Katja Fieler. And I give the stage to you, Ezgi. Please carry on.
Thank you. Thank you very much. So first I'm going to share my screen. Okay, I hope everybody can see my screen right now. Yeah, good. Okay, so hello everyone. Today I'm going to talk about an fMRI study on tactile suppression. And more specifically, the study investigates how reach-relevant somatosensory signals modulate neural activity associated with suppression. Before that, I would like to give you a very brief intro on what tactile suppression is and how it can be measured. So tactile suppression is the phenomenon that our sensitivity to tactile signals on a moving limb is reduced. It can be observed, for example, for self-produced tactile sensation. So here in this example, the participants are asked to produce a presented target force. And when they produce this force themselves, they match it with a higher force than when they produce it through a slider. Tactile suppression can also be observed during reaching movements and with external applied tactile stimulation. So here in the study, the participants are presented with a brief vibrotactile stimuli on their index finger in various phases of the reaching movement. And what the researchers observed was that the detection for these tactile stimuli was way lower right before and during the course of the movement. Okay, so it's thought that suppression occurs as a result of an internal model that predicts the sensory feedback of the movement and attenuates these signals that match the predictions. And this is thought to compensate for our limited capacity to process more relevant or novel sensory information. And such a processing also requires an efficient weighting and integration of available predictive and feedback signals. So when we look at the neural implementation, it's also in line with what we see in behavior. So tactile suppression has been found to be associated with reduced activation in somatosensory areas such as the primary and secondary somatosensory cortices, S1, S2, as well as the insula. Activation changes have also been found in the premotor cortex and more specifically the supplementary motor area that is implicated in the planning of intentional movements and generation of motor commands. Also, deactivations have been found in the posterior parietal cortex, including the inferior parietal lobule, the supramarginal gyrus, as well as the cerebellum. And these are areas that are strongly linked with the prediction and monitoring of movement-related somatosensory signals. So we have a clear picture of what tactile suppression is and its neural implementation, but more recent studies have demonstrated that it's not an all or none phenomenon. So we have the strength of suppression can actually be modulated depending on the relevance of the somatosensory feedback signals. So for example, suppression is greater when we reach to our own hand than we reach to an external target. And this is possibly to upregulate the reach-related processing on the target hand. Likewise, we also see greater suppression when feedback signals are less important. So when we are about to grasp something, suppression on our index finger that would grasp the object is actually less than on our forearm. So there are definitely these modulations. So there is a variety of behavioral evidence, but the neural implementation of this modulation is not clear. And in this study, this is what we investigated. So we specifically asked the question of whether bold activation associated with suppression is modulated by the relevance of the somatosensory feedback signals. And for that, we used reaching movements to one's own hand or to an external target. And we expect modulations in regions associated with the processing, integration, and prediction of tactile stimuli during suppression. So we would expect modulations, changes in modulation across these two different movements. So here's what we did in the experimental paradigm. The participants are asked to reach either to their own left index finger, so that would be the somatosensory reach goal, or to a touch screen. This is the external reach goal. They were also, they also received a brief tactile vibration on the to-be-moved index finger. That was to probe suppression. So we have probe trials. We also have trials without any stimulation. And importantly, we presented the probes right before reaching movement. So this would be the planning phase, and when suppression can be measured without the interference of the movement itself. At the end of the reach, we asked them whether they detected anything, and they answered either yes or no. So this is how movement trials look like. We also presented baseline trials in which they received same stimuli, but this time without moving their hands. So in this case, we shouldn't be seeing any suppression. Okay, so here are the behavioral results. So here on this graph, you see a proportion of detection responses as a function of stimulus intensity. We presented stimuli of different intensities. And we also fitted psychometric functions to the proportion of detection responses for each condition in order to get a detection threshold estimate. And here you can see that, so this is one participant, that the detection thresholds for the baseline condition is much lower than thresholds for the reaching conditions. And this actually confirms this suppression effect. We also looked for differences between the different reaching movements in terms of suppression by obtaining a threshold difference. So this we did by subtracting each threshold, movement threshold, from the baseline condition, and we compared these two. And we saw that there was stronger suppression when participants reached to their own index finger, which is in line with our expectation. Okay, so here are the behavioral results. In order to assess bold activation associated with suppression, we focused on the interaction between movement, so these different movements, and whether there was a probe presented or not. So in this condition, you see we contrasted external with somatosensory reaching movements, and also for probe present and absent trials. And these are the results. So we see that the activation in the SMA, the supramarginal gyrus, insula is modulated by whether the movement is somatosensory, whether the goal target is somatosensory or external, and also whether a probe was presented or not. So this you can see more clearly by just looking at the activations in, so here are two examples, two regions of interest. So this is the postcentral gyrus S1 and the SMA. And you see that it's modulated by whether a probe was presented. And in the somatosensory condition, we see lower activation in these areas. We also used PPIs to assess changes in coupling between different areas that are associated with the processing and integration of somatosensory signals. For this, we chose the supramarginal gyrus. Supramarginal gyrus has been shown as an area that is implicated in the integration of somatosensory signals. And we assessed functional coupling between this area and the S2 as well as the insula. So these are somatosensory processing areas. And we found that this functional coupling was stronger when the reaching movement was somatosensory. And again, when there was a probe presented. Okay, so to sum up, we in our study, we confirmed that tactile suppression is modulated by the relevance of the somatosensory feedback. We also had bold activation that is in line with what we see in behavior. So we observe that probes during somatosensory reaching is associated with deactivations in areas that are involved in somatosensory processing, such as the S1, the insula, as well as somatosensory integration and prediction, such as the supramarginal gyrus, and also planning, movement planning, and motor command generation. And this would be the SMA. And increased coupling between the supramarginal gyrus and S2 as well as the insula suggests that the modulations in integration of somatosensory signals are seen for different types of movements. And to sum up, overall, our results provide the first neural evidence that suppression is not an all or none mechanism and can arise from flexible modulation of predictive and feedback signals during reaching. And that would be the end of my talk. I would like to thank my co-authors and also student assistants that helped with data collection, and thank you all for listening, and I'm happy to receive questions.
Thank you so much, Isgai. Do we have any questions? Yes, we do have one question from JJ Orban. The question is there any way to look at processing integration movement separately, because those are very different processes?
Can I have the question again, or maybe I can also check. Okay, is there any way to and movement separately?
You got it in the chat?
Yeah, now I can see that. I agree totally with that. It's really difficult. So at least in this case, we try to account for movement-related effects by having the probe present and absent trials. So here we focused on probe present trials, and in probe absent trials, the idea is to at least account for movement-related effects. But yeah, I agree. That is quite difficult.
Okay, thank you so much. And thank you, Urban, for the question. Do we have any other question? Any discussion, any query with others? Okay, so it was really a very interesting talk, listening to tactile suppression. It was really very interesting. Thank you so much, Isgai.
Thank you.
And,
thank you.
We now move to the next session, the next speaker, Alexandra Herman. Alexandra Herman is a postdoc at Royal Holloway, University of London. Next week, she will be moving to NENCA Institute of Experimental Biology. And all the best to you, Alexandra, for your new start. And it's up to you now.
Thank you very much for hosting this session, and thank you to everyone who's attending. I'll be talking again about somatosensory detection and how it may be different or similar to interoceptive processing. So first, a little bit of background. So bodily self-consciousness is the identification with one's body, self-location of body and body parts in space, and the first-person perspective. And all of these processes depend on the perception and awareness of bodily signals. Importantly, it also depends on the brain's ability to integrate information about the body originating from different sensory modalities, such as somatosensation, proprioception, and also interoception. However, little is known about how these apparently different divergent signals converge in the brain to form a bodily self-consciousness. And particularly in that topic, interoception has received a lot of attention recently. And interoception is the sense of the physiological condition of the body. And although it is related to all systems involved in maintenance of homeostasis, such as respiration, cardiovascular system, gastrointestinal system. The cardioception, so the perception of one's heart, received the most attention in that topic. And there are two commonly used ways of measuring interoception. The one is heartbeat counting task, in which participants are asked to silently count their own heartbeats as they feel them in predefined periods without manually checking their pulse. Another way of measuring interoception is the heartbeat discrimination task, in which participants are presented with a series of tones and they have to make judgments whether these tones are in sync with their own heart or out of sync. And as you can imagine, the second task is much more difficult. Regarding the neural correlates of interoception, most of the studies focused on the aspects of interoceptive attention, whereby participants are asked to attend or monitor their heartbeats, and this is compared to a condition when participants are asked to attend to external stimuli, usually visual or auditory stimuli. And these studies have shown that interoceptive attention compared to exteroceptive attention engages a widespread network of frontoparietal and occipital regions, which also include the insular cortex. And insular cortex is considered a particularly important role in interoception in a study that looked at the aspects of interoceptive accuracy, so our objective ability to perceive heart well, accurately. The study showed that better performance in the heartbeat discrimination task was related to higher activity in the right anterior insular cortex. Therefore, it is believed that right anterior insula is a hub for interoceptive processes. But those past studies mainly looked at the neural correlates of interoceptive attention and to a lesser extent accuracy between internal stimuli and external stimuli. Those paradigms used did not allow to consider the neural correlates of conscious perception of those internal stimuli. And also it is important to note that typically interoception is compared to exteroceptive distal stimuli such as visual or audition. But it would be important to compare interoception to more proximal body-related stimuli such as somatosensation, as we know that the two of them play such important role for body self-consciousness. So for this reason, we developed a novel cardiac and somatosensory detection task. In this task, participants were first instructed to focus either on their heartbeat or faint vibrotactile stimuli applied to their left hand. And following intertrial interval, fixation cross changed color 3 times. And this change in the color of fixation cross was a period where participants had to perceive the heartbeats or somatosensory stimuli. And next, participants had to indicate with the corresponding button press or multiple button presses when they felt the stimulus. And finally, they also rated their confidence. So this paradigm allowed us to differentiate between the focus, so attention to stimuli, and detection of those stimuli. For this reason, we performed 2 different analyses. First, focus on the looking at the attention to heartbeats or somatosensory stimuli, including this whole period of all 3 colored screens. And for the detection analysis, we separated each color for cross and labeled it according to signal detection theory as hit, miss, correct, rejection, or false alarms. So if, for example, someone reported that they felt the heartbeat during the red cross duration and there was indeed a heartbeat there, that was considered a hit, and so on and so forth. And participants also performed a heartbeat counting task to correlate the performance in our novel task with this more standard version. In terms of behavioral results, we find that accuracy between cardiac and somatosensory condition was very much comparable. There were no significant differences there, and it was also quite similar across blocks. There were 4 blocks for each condition, 20 trials each. The situation was slightly different for the confidence ratings. Participants reported higher confidence level for the somatosensory versus cardiac condition, which may indicate that even though looking at the accuracy, the objective difficulty was quite similar, participants might have perceived the somatosensory condition as slightly easier. We did not find any significant correlation between performance on the heartbeat counting task and our new detection paradigm, but the cardiac detection accuracy correlated with the somatosensory detection accuracy, which may be related to the similar difficulty of both tasks, but also obviously underlying neural correlates. Regarding the neuroimaging results, first we looked at the main effect of cardiac and somatosensory focus. So regardless of the accuracy, how they perform, just when they had to focus on the stimuli. And what we see here is that for both focus condition and somatosensory condition, quite a robust network of frontal, parietal, and occipital regions got activated. And that also included regions typically reported in interceptive research, such as somatosensory cortex, insula, and so on and so forth. But importantly, our conjunction analysis shown that there is a huge overlap between those, between the focusing on someone's heart versus stimuli applied to one's hand. Despite this overlap, there were also some significant differences. That is, cardiac focus condition evoked higher activation in bilateral superior frontal gyrus and lateral occipital cortex compared to the somatosensory condition. So both of these regions are related in attention and also frontal gyrus in cognitive control. So it may reflect higher attentional resources needed in order to focus on one's heart. That's at least how we interpret it. Next, we looked at the main effect of conscious detection of stimuli, both for cardiac and somatosensory conditions taken together. And what we see, hits versus misses evoke this very robust activation pattern, including frontal, parietal regions, also subcortical regions, caudate, putamen. We have insula there as well. And if you look at this, it is very similar to the salience network. And obviously, salience network is important for switching between internal and external oriented attention. That's why it may be related to higher activation during hits versus misses. The reverse contrast, misses versus hits, evoke not as a large, robust activation, but we see some prefrontal cortex areas here, parietal cortex, and also temporal cortex, and we have hippocampus, temporal cortex, cuneus. So this is much more similar, or bears at least some resemblance to default mode network, which is related in task of activity, but also self-referential activity. So this activation pattern that we see here may be related simply to inattention, but also focusing inwards to oneself and on one's body. We did not find any interaction between conscious detection of cardiac or somatosensory detection stimuli. That's why it suggests that this activation may show large overlap between those two conditions. And indeed, for hits versus misses, we see that there is a large overlap for cardiac and somatosensory stimuli, including subcortical regions, but also temporal and parietal areas. For the hits versus misses, we see prefrontal and parietal activation, but there was no significant overlap, suggesting that there might be some differences there, but it did not reach significance for the interaction effect. And finally, as insula cortex is considered this key hub for interoceptive processing, Whoops, sorry. We also did a PPI analysis, and just because I'm running out of time, I'm just telling you, I just wanted to tell you that there were significant differences here for the connectivity, which were driven by the cardiac condition. So this is my summary, and if you want to read more, there is a preprint available online. Sorry for going out of time.
You're perfectly all right. Thank you, Alexandra. It was really a very wonderful talk. We have a question from Sky. Is detection accuracy for cardiac focus below chance, or is it the usual finding?
For the heartbeat counting task, there is no performance below chance. It just was very low. For the discrimination task, when you have series of tone and then you have to judge whether it's in sync and out of sync, you do see many participants who perform below chance, which is quite weird. But we think it's because some people feel their heartbeat or have this maximum time when they feel their heartbeat, not necessarily on systole, but someone else somewhere later in the cardiac cycle, which is not involved in this pattern, which we usually use for this type of task.
We can accommodate one more question because we're running out of time, and then we will have questions later on in the case we have time. The question is from Rebecca. What kind of somatosensory stimuli did you use?
It was a faint vibrotactile stimuli, so it was basically a sound wave converted into a puff of air, which is very faint.
Okay, thank you so much, Alexandra. It was really a wonderful talk. There are a few more questions which are there, so we will try to do it up right now. Otherwise, I will suggest the audience to connect with you directly on your Twitter and ask there as well, right? Okay, so now we move on to Kai-Ching Kong. She is currently a postdoctoral researcher fellow funded by Faison Foundation Fellowship. She is working with Professor Alexandre Farnay at INSERM U1028 CNRL, France. Before that, she worked with Professor Patrick Haggard at Institute of Cognitive Neuroscience, UCL, UK. And her research interest includes sense of agency, sensorimeter control, body representation, peripersonal space, and tool use plasticity. Kai-Ching, please continue.
Thanks, Amita, for the introduction. I'm Kai-Ching. As Amita said, I'm currently a postdoc at INSERM Lyon. But the study I'm going to present is a study I did at UCL recently before I moved to France. So the title is the interhemispheric communication during haptic self-touch perception. First, a bit brief introduction. We know that bimanual interactions are common. Successful bimanual activities may require transfer of both sensory and motor information between hemispheres. In haptic exploration, bimanual information might be superior for several reasons, including we have more and redundant information. On the other hand, intermanual interference may occur. For the integration, hemispheric limitation of bandwidth applies to simultaneous perception, but not so much to the second type of stimuli. Previous data are equivocal as to whether there is a interhemispheric integration or not during the bimanual haptic processing. So here we want to ask what information is shared between the hemispheres in haptic self-touch processing with the second type of stimuli. In our study, the self-touch stimulus is based on stroking over a positive asperity, which is a bump, with one's own right fingertip on the left arm. Just imagine something like this. Close your eyes and use your right hand, move on your skin, and you encounter a bump. So this bump perception is signaled by two cues. One is kinesthetic information on the right hand, on my right hand, and the other one is tactile information on the left, on my left hand. In our study, is the the tactile information on the left arm. In our study, we decapped the congruence between the two cues with a force control robot system. So the kinesthetic cue will be always perceived as a bump on the right hand. And the tactile force cue could be a bump or a hole based on the design. So we want to know whether and how the tactile information on the left arm integrates with the kinesthetic information on the right hand. To do so, we developed the novel setup for the self-touch stimulation, as you see here. It's a master-slave robot system. The right robot is a master robot, and the left one is a slave robot. So the slave robot reproduces any movement from the master robot, and the form board here is to block the view of the robot's and participant's hand. And you see participants place their left arm and hand in the module support, and use their right hand holding the master robot, made movements on the flat surface here. During the movement, resistances were added to generate elusive bumper perception. Simultaneously, the left robot stroked on participants' left forearm with a paintbrush here, gently stroke on their left forearm. Resistances were manipulated to generate a bump, a hole, or a flat stimulation, which might be different information from the right hand. So I'm going to show you two short videos to make you more clear about what I'm seeing here. So one is for left hole stimulation, in which participants had a decreased pressure experience, and the other one is left bump stimulation, in which participants had an increased pressure experience. Please pay attention on the paintbrush movement on the left forearm to see how the pressure changes. So first, two movements in each trial. Here, the first is a flat stroke. The second is a hole stimulation, it's a pressure decrease. And for this one, the first stroke is a pressure decrease, and the second one is a flat stroke. As you see here, the right and the left robot are temporally synchronized, which is not influenced by the bump or the hole manipulation. Let's see more. Let's see the experiment design. We have two control experiments and two main experiments. In the control experiments, the experimenter moves the master robot, and the participant just passively feels the tactile stimulation on their left arm. Our goal here is to check whether the people can perceive the difference between the bump and the hole on the left arm to validate our manipulation. So in control experiment 1, 2 strokes in each trial with 4 possible combinations. And after each trial, participants were asked, were the sensations of the 2 left strokes same or different? And in experiment 2, only 1 stroke for each trial. And the participants were asked, did the pressure on your left arm show an increase or decrease? I'll just briefly tell you directly tell you the result. Yes, they can. Let's move to the main experiments. In main experiments, participant moves the right master robot and feels the tactile stimulation on their left forearm. We use the 2FC task and a constant stimuli method. A reference in each trial, there is a reference and stimulus and a test stimulus. In the task, participants made 2 movements with the right hand and judged which movement felt more bumpy, first or second. The tactile stimulation is, so here the left tactile stimulation for the judgment is task irrelevant information, we use the block design in experiment 1 and randomized design in experiment 2. Let's see more details of the design. Don't be panicked by the slide. Let's see what the right side first. So what happened with the right hand is same for experiment 1 and 2. So 2 movements in each trial. One is a virtual bump represented by the dashed line here, which is a reference, and the other one is one of the 5 possible virtual bumps here, which is a test stimuli. And the order of them is randomized. So let's see the left side. For experiment 1, we have 5 blocked conditions: bump first, bump second, hold first, hold second, and flat condition as a baseline. Let's take bump first as an example. So 2 strokes in each trial consist with what happened with the right hand. The bump first means there is always a bump tactile stimulation for the first stroke, and the second stroke is a flat one. And for the bump second, the bump stimulation is on the second stroke. It's same for the whole conditions. In experiment 2, we randomized bump first and bump second, so it's bump randomized, which means both the first stroke or the second stroke can be bump tactile stimulation or flat one. It's same for the whole randomized condition. And the flat condition here is the same with the condition E in experiment 1. So you can see here, the left bump, left hold, and left flat stimulation can be added to the reference or the test stimuli in both experiment 1 and 2. So we have 5 conditions of 5 conditions: left bump to the reference, bump to the test, hold to the reference, hold to the test, and the flat condition. Please remember this. We're gonna see this in the result. So as we have 5 different test stimuli, we fitted the average proportion of the test is bigger as a function of test reference difference onto the psychometric curve. Let's see experiment 1 first. So the middle black curve is the flat condition, which is a baseline, and the other colorful curves shifted from the baseline, which means the bump or the whole tactile stimulation on the left best, the judgments on the right. Let's see an example. The orange curve is bump to the test, which means adding a left bump to the test makes the right bump feels more stronger, which means more bumpy. Similarly, the green curve here means adding a hole to the right bump also makes the right bump more bumpy. It's a similar thing for the other 2 curves. In experiment 2, we still can see some shifts here, but the curves are almost overlapped in experiment 2. From the psychometric curves in experiment 1 and 2, we can get PSE, which is the point of subjective equality, and the slopes, which is an indicator of the sensitivity of the discrimination. So let's see the result of PSE and slopes for the within group analysis first. So the larger the PSE is, the more likely it becomes that the test stimulus is perceived as being smaller, which means less bumpy than the reference stimulus. We performed 2x2 ANOVA on the bump hole and the test reference. The main effect on the test reference and the interaction effect showed that judging a bump with the right hand movement is biased by the left tactile stimulation, which means both left bump or left hole makes the right bump feels more bumpy. So it's not a sound integration, which means not integrating the full geometric profiles. If that's the case, adding a left hole to a right bump will make the right bump less bumpy. And so in experiment 2, even we see there is a significant difference between.
Can we come to the summary? We have limited time, please.
Okay, perfect. So we performed the same analysis for slope. We did not find any significant difference, which means participants' sensitivity for the discrimination of the right bump was not influenced by the left tactile stimulation. And let's see the across experiment analysis. We added the bump. We added the blocked or randomized design as one more factor to perform the ANOVA, and we see that the bias, in fact, only occurs when there is a regular relation between the right kinesthetic information and the left tactile information, which is a blocked design, not the randomized design. So we concluded here the tactile force profiles consist with either positive or negative asperity produced a bias to perceive a positive asperity with the right hand. However, the bias doesn't reflect detailed sound geometric information about the tactile forces. So participants are just learning a constant temporal relationship between two events. However, they are not combined as they were parts of a single spatial percept. And compared to previous studies, so the transfer between tactile information and kinesthetic information are not symmetric. And back to our question, what information is shared often is provide evidence that tactile temporal information, but not tactile spatial information is transferred interhemispherically to integrate with kinesthetic information. And this integration requires that participants can form a model of a regular relation between the left and the right events. Okay, that's the end of my talk. I'd like to thank my advisor, Patrick Haggett, and my colleagues from UCL, and especially Miruna helped a lot with the data collection, and a collaborator from NTD and the funding agency. Thanks very much for listening.
Thank you so much, Kai-King. It was a wonderful talk. Do we have any questions? I think we can just squeeze in one question from any of the audience.
I'll see the question.
We have just time enough for one question. Any audience would like to ask? Yeah, so there is one question from Patrick. People need the regularity of a blocked design in order to integrate information. But what they are integrating is just temporal information, which should be present in the randomized condition also. So it looks like people need regularity to do temporal integration, even though temporal information is always there. Can you comment? You can see the question in the chat.
Yes, I'm trying to look at it. I'm clear with what Patrick said on the first part, but at the end of the question is to
he's asking for your comment on this particular aspect, basically.
Okay, I see it. It looks like people need regularity to do temporal integration, even though temporal information is always there. Yes, I think in randomized design, the temporal information is right, it's also there. But as we have randomized left tactile stimulation, maybe participants have they lack the capacity to form a model or learn the relation between the left and the right. So they cannot integrate. I'm not sure if it answers the question. Thanks.
Thank you so much. Thank you so much, Kai-King. Thank you, all the panelists. Azgai, Gaiking, and Alexandra. It was really a wonderful session. Thanks to all the attendees for their interaction and the questions. And definitely a special thanks to our backend support, Sophia. Thank you, everyone. And let us see you in the next session. Okay, bye.
Thank you.
Bye.
Thanks, Amita.
Thanks.
Bye.
Saying if you can ask the question in Q&A box. So there is a, not in the chat, but in the Q&A box. So if you can type your question there, then we can actually vote it up and things like these. Okay, so we have Vinicius Carvalho coming. Hi, Vinicius.
Hi, how are you?
Hi, yeah, so I mean, if you like, I can stop sharing my screen, and if you like to do a quick check with sharing your screen and see everything is working for you.
Yeah, okay, try it. Okay, just a second. Okay, can you see that all right?
Yeah, that looks good.
Okay, just the presenter modes.
Yeah.
And you're seeing only the presentation, right?
I can see, yeah.
Okay.
I only see your presentation. I mean, you are not in full screen mode yet, so.
Is it not appearing as full screen?
I mean, you're not at, so there's, yeah, so you're not in full screen. So.
Yeah, you have to go on F5 presentation mode, then it's going to go on like full screen.
Oh, there's a.
I'll try to remove.
Oh, there is a, yeah, that's great.
Okay.
Yeah.
Probably was the another monitor I was using.
That's good. Okay, so that's fine. So I will start. Yes, so maybe I could just share the screen again.
Okay. Can you see and hear me okay? Was having trouble with the connection
no,
here?
It's all great. We can see you, we can hear you. Your slides are sharing well, so
okay, cool.
It should be all great.
I hope it stays that way.
It will. Okay, so so there's still a couple of minutes, so we can start in a couple of minutes. Yes. So for Vinicius, you have 12 minutes. So there are some instructions on the screen. So and all the participants, if you can, again, there's nothing like a dumb question. So if you have a question, type it in the Q&A box, not in the chat box, but in the Q&A box. Okay. And you can, once you have questions there, you can vote it up. You can do a few things there. So please ask questions when, and then when the talk finishes, we can have those questions answered by the panelists. So please do ask questions. You can also chat to panelists or everyone. So there's a chat option there. Use this as well as much as you like. That's great. And we have Vinit coming and joining us. Hi, Vinit. I saw Vinit. So Vinit, I think I have promoted you, Vinit, to panelist. So yeah, Vinit, are you there? Can you hear us? Hi, Vinit. Yeah, you're on the mute. So can you quickly, because we have to start within a minute. So could you please share your screen and see everything is working for you?
Yeah.
And then Rodrigo will share the screen. So
Can you see me?
yeah, we can. Okay, so you have something else, but that's all right. So the screen is sharing. Okay, that's great. So it's over to you, Rodrigo. So we have the first talk given by Rodrigo Secura de Souza from University of Sheffield, and he is going to talk about foot sim, modeling tactile responses from the sole of the foot. It's all over to you.
So yeah, hello there. As I was kindly introduced by Adele, my name is Rodrigo. I work at the University of Sheffield as a research associate. And today we're going to be talking about the tactile responses of the foot sole. But why are those tactile responses important? Well, they're important mostly for gait and for balance. So when we have a normal subject on an experiment walking and we use anesthetics on their foot sole, like we can see on those plots over here, the velocity of gait, which is the y-axis of the plot, gets significantly reduced, as you can see at the gray bar in opposition to the black bar, when you anesthetize the foot. The opposite is also true. If you have patients with suffered stroke or they have diabetes, if you put vibrating insoles on their shoes, you really reduce the postural sway that you have on those conditions. So the tactile feedback from the foot sole is very important for walking and gait. But where does it come from? Where does information come from? Well, most of this information that we have for tactile afferents came from the hand, but we have 4 classes of tactile afferents. They are basically divided in how fast or slow they adapt to stimuli. So you have slow adapting and rapid adapting, and you have types 1 and types 2, depending on the sizes of the receptive fields. So small receptive fields type 1, large receptive fields type 2. So you can see on this color image here, the layers of the skin, the epidermis and the dermis, and where those receptors are placed. You have the Pacinian corpuscles, which are the RA2s over here, and the Messina corpuscles, which are the RA1s, which are thought to be the ones that have most importance for tactile feedback on the foot sole. But how do you study those afferents? Well, one way is to use microneurography, like our collaborators Leah Bent and Nick did on Guelph University. They got participants to sit on and get electrodes implanted on their nerve, and then they recorded one afferent at a time. It's actually a very hard to do technique. It takes time. The subject has to be immobilized, and then they applied a range of amplitudes and frequencies of stimulation to the sole of the foot, a range that you would have in normal conditions like walk, from 0.01 millimeters to 2 millimeters, and frequencies ranging from 3 hertz to 250 hertz. And then they recorded this data set. So using that data, we built a model that takes into consideration the specific skin mechanics of the foot and deliver pressures to a quasi-static model of the skin that then is given to integrating fire neurons that have 12 parameters that you can interchange, a low pass filter. All of those 2 to 7 are time varying parameters. And then you integrate all of those into a saturation parameter because of the adaptation of those tactile afferents. Then you have a couple more post-inhibition parameters to account for refractoriness of the receptors. Also, as I mentioned, the skin of the foot is not like the skin of the hand. It's not homogeneous. So you have, as you can see on this blue and green foot sole plots, you have harder regions, which are the darker colors, and softer regions, which are the lighter colors. The same goes for thicker regions and thinner regions. So the heel is always the harder and thicker region versus the archer, which is softer and thinner. Also, the density of each one of those afferents varies in the skin, and this data also came from the microneurography study. So you have the density for each one of those afferent classes. So then we put the model to the test. Is our model firing the way those afferents were firing in the microneurography study, in the empirical one? So in this plot, you have on the y-axis the way our model fired for the combinations of amplitudes and frequencies that they did also on the empirical experiment. And on the x-axis, you have the firing of that experiment per se. So you can see that for the FAs, the top ones, the blue and the orange ones, as you can see in the legend, we are pretty much in the ballpark of what they should be. The assays are not so good. The reason for that being that for SA2s, we only had very few models to use to optimize the parameters on the fitting procedure that we did using differential evolution. And also our model doesn't account for stretch as well, and that's the main thing that SA2s are known for, one-off. So putting that to the test, we also went to the thresholds, different firing thresholds, which are the minimum amplitude per frequency that lists a certain firing rate, a certain number of spikes. So here you have on the y-axis the amplitude, and on the x-axis the frequencies of each one of the models, which are the individual lines, and the thresholds. Top 4 panels on the model, bottom 4 panels, the empirical ones. So they look quite similar. So we were pretty happy, and oh yeah, the model is behaving like it should be. So let's clump those models, group them per class. So if we group those per class, you can see those two graphs on the left. On the left, they are panel A is what came from the microneurography experiment. So we can see those thresholds there for each one of the classes, same color code as before. Panel B is our own model. And then we looked and said, they look very similar. So we went and measured the RMSAs, which is a measure of difference in between those two curves. And if you look at this graph on the right, you can see that the FAs, 0 there mean the curves are really similar. We are in the optimal scenario, and 1 being the worst possible case, that the FAs behave much better than the SAs, and they are all in a good range. The scatter points are actually the individual models over there. So it seems that everything is going well, that the model is behaving the same way as the afferents on the actual foot of both subjects behaved. So we decided to go even further and put the model to the test. We had subjects on the Insignio Institute done by our collaborator Claudia, putting sensorized insoles on their shoes, which are basically pressure sensitive insoles that can get pressure off your feet when you're walking or when you're doing any activity. And then using those pressures, we converted those pressures into indentations. That's what our model used, because as you remember, microneurography can only be done with patients immobilized and one afferent at a time. So now we can take these pressures and turn them into indentations, feed them back into the model, and do experiments that would not be possible in that scenario. And then we found out of the bat that for all the phases of the step, like the initial contact, the flattening of the foot, the mid stance, and the push off, you have different activations of each one of those afferent classes. So if you look at the bottom of the image, you have 2 foot soles paired. The red ones are the heat maps of the pressure sensitive insoles. So you can see how the pressure is distributed in each one of those parts of the step. The other foot, the one in black, is the activation of the actual neurons, the actual afferents. And we can see that on the initial contact, there is a burst of FA2 firing, which was something that was not known. Granted that it was a proof of concept experiment, was not that well controlled, but is a very good indication that we can actually use the model to experiment in ways that wouldn't be possible using microneurography. And then just to show you a small animation of that step happening, same as before, the heat map of the pressures on the left-hand side and the activation of the mechanoreceptors, the tactile afferents on the right-hand side. And you can see here again, the plot of those firing rates per time, and you can see the bursts in FA2 firing in the initial phases of it. And then this model is already being applied to prosthetics development by our collaborators on ATH Zurich, Natalia and Stani. So they were cover of Nature a while back. They are doing real feedback prosthetics by having sensorized insoles, giving stimulus to the nerve by using implants. And then using the things that we are optimizing the now model, we aim on giving actually a good account of what the sensation would be for each one of those tactile afferents in the foot sole and restore the sensation on those subjects. So that's basically it. This is the team, Hannes, myself, and Luke and Claudia worked on the insoles, the guys on the ATH, Natalia and Stania, and Nicholas and Leia that did the microneurography experiments. So thank you very much for watching, and you can fire any questions you want.
Okay, so that's great. So thank you. That was perfectly on time. And yeah, so please ask any questions by typing them into Q&A box. So anyone who has questions, panelists or the audience, please type this into Q&A box. While people are thinking about it, maybe I could start. So I probably missed it. So what was these? So you had 4 different FA1, FA2, SA1, and SA2. So these are 4. So what they were, how they were different from each other? So what they're measuring?
So we got the data from the microneurography experiments. And then our model has the integrating fire neurons with 12 parameters. So we applied differential evolution to those models to try to fit them to the firing or each one of the afferents that were recorded in the experimental setting. So basically the same number of afferents that they recorded, we have it as models. So the total was about 52 models divided per class, a certain number of them in each of the classes. And then that's why I just showed you a representative image. Otherwise, I would show you a massive scatter plot full of information.
Yeah, of course.
Then I clumped them into classes. That's why you only saw 4 classes, but those 4 classes actually came.
And how do you do that? Did you run some sort of a dimension reduction, or?
Sorry, I didn't catch that.
So how did you do that? So you said you had quite a lot of different, bunch of things, and from this you pull this out. So how do you do that?
So we had the firing rates for those experiments. So we knew that amplitude 2 and frequency 3 hertz elicited 50 hertz firing rate. So we had the responses of those afferents, and then we threw them into a machine learning differential evolution algorithm to basically tell me which combination of the parameters on that differential equation, which is the leak and fire model, would provide me with a response that was closer to the actual empirical one. That's how we did it.
Okay, that makes sense. Okay, so that's great. So I think we are going on time. So thanks a lot, Rodrigo, again. It was great talk. You're welcome. And so next we should crack on. We have a next exciting talk, which is from Vinit Trivedi. And he is MD PhD student at the School of Medicine, Emory and Georgia Tech. And he's going to talk about network oscillations in antidepressant deep brain stimulation. So over to you, Trivedi.
Great. Thank you so much. Thank you for the introduction. I'm Vineet. I'm excited to present today on network oscillations and antidepressant deep brain stimulation. Did you all see the slide just change?
Yes, I think, but we do have another slide on top.
Do you have the notes on top?
Yeah.
Okay. Let me try it one more time. Sorry about that. I thought I selected just the window. Do you still see the notes?
It's coming. Yes, we still see the notes. So we can't see your slide. I mean, you could have, no, that's better. So we have your slides.
Can you see the
Yes.
slide right now?
Yes, now we can see that.
Okay, that's fine. I have another computer over here. All right. So thanks again. Sorry for the technical difficulties. In the next 12 minutes, I'll review DBS, its use in depression, and the reverse engineering paradigm we're using to study it. I'll talk a little bit about the major results from our empirical work, most of the work that I did during my PhD, before discussing the biophysical modeling efforts and future directions for the field. Okay. First, the work is a result of a huge team effort, and I want to thank everyone that helped, especially the support from Whitaker Foundation and Emory MSTP, and a special thank you to the patients who ended up being crucial collaborators in the effort. So, deep brain stimulation is a neuromodulation therapy involving the implantation of electrodes into specific parts of the brain. In this picture, that's implanted into the subthalamic nucleus. This enables a neurostimulator to deliver therapeutic patterns of electrical currents, typically around 130 Hertz, into brain targets associated with disease. Implanted DBS leads have multiple electrodes available, with at least one electrode being near a desired target and the stimulation voltage being a parameter that can actually be used to adjust how much electricity is being delivered to engage that target. Now, DBS is commonly used for motor disorders, but promising applications in psychiatric illnesses are growing, with multiple brain regions and multiple disorders being studied. Today, I'm going to focus on subcallosal cingulate, brain target, and depression. Depression is a psychiatric disorder characterized by persistent sadness, sleep difficulties, suicidality, and other non-specific symptoms. Neuroimaging studies have actually helped identify a network of brain regions associated with depression. One node in particular, the subcallosal cingulate CG25, it plays a central role in depression, being metabolically hyperactive in depression, and hypoactive when successfully treated. So the picture above and below show how SCC changes its metabolic activity as a result of depression state. Now, DBS in the SCC showed early efficacy in treating depression, but recent advances and refinements in our lab have actually specifically implicated white matter tracks, the axons in the brain, as the necessary therapeutic targets, specifically for white matter tracks, the uncinate fasciculus, the forceps minor, the cingulum bundle, and the thalamic fibers. Here you can see each one colored in a different color. Now, this was a retrospective study. In a subsequent prospective study where individual tractography was targeted in each patient, individualized, we demonstrated an improvement in the efficacy, which implicates a little bit the subcallosal cingulate white matter is actually the therapeutic target we need to hit. Now, this SCC white matter DBS is what we currently do. We actually specifically target white matter tracks and not the broad gray matter of the SCC. So this is essentially what our group performs today, and we're continuing to study it. Now, how do we study it? Because while SCC white matter DBS appears to work in treating depression, its mechanism of action remains unclear. Modern efforts in DBS are moving towards adaptive strategies that rely on measurements and algorithms instead of physician assessment to optimize DBS stimulation. The clinical devices that have been developed give a unique opportunity to study how DBS changes brain activity and how brain activity is linked to behavior. Now, using a prototype DBS device capable of both stimulating and recording in patients, in this case, the Activa PC+S, which came out originally in 2013, we can actually measure local field potentials multiple times a day over years in our patients that we're treating. The results I'm presenting today are from 6 patients implanted with the PC+S between 2013 and 2018 who have all successfully recovered from depression. Because of the clinical nature of this work, we had to rely on several different approaches to study our therapy, including benchtop testing of the PC+S, intraoperative recordings, extraoperative local field potentials multiple times a day, and monthly experiments with combined LFP and EEG. Now, because of the small sample size, we had to take a different approach, one built on models that the clinicians already use to treat patients and rely on the data that we're able to collect, sparse and noisy though it may be, to refine those models that the clinicians are already using. So our first goal, as every good first goal should be, is characterizing our measurement. How good are our measurements? And in particular, our PC plus S recording had this issue called mismatch compression. And we had to correct for that before we could actually analyze the brain oscillations we cared about. Then we wanted to study how the brain oscillations actually related to behavior to identify a depression readout. Can we actually predict how depressed a patient is from the oscillations we can record? And then finally, we wanted to study how DBS modulates the physiology of the brain. And this gets at characterizing the specific network action of on-target SCC white matter DBS instead of broader SCC regional stimulation. I won't talk much about mismatch compression in this talk, but suffice it to say we developed a pre-processing strategy to remove the impedance mismatch caused artifacts in our chronic SCC LFP recordings. And this enabled us to actually reliably study oscillations in the brain. So I'll focus mostly on 1 and 2. Diving into 1. So to identify SCC oscillations related to depression, we implemented our therapy exactly how we implemented it before, which is implanting the patients, turning the stimulation on 1 month after the implantation, and then following them weekly to assess how depressed they are. These are the Hamilton Depression Rating Scales, our way of measuring depression over the course of about 8 months. And higher corresponds to worse depression. In parallel, and blinded to the clinicians, I analyzed LFP recordings that were measured approximately once every 4 hours over the course of 7 months. And these we downloaded every week and enabled us to actually have LFP recordings, multiple LFP recordings a day and about 70, 80 recordings a week associated with each of these weekly Hamilton Depression Rating Scales. This data set was split into a training and a testing set, about a 60-40 split, with each recording associated with that week's depression score. An elastic net regularized linear regression model is trained through a nested cross-validation approach before it's assessed in the 40% training set. And the 40% training set performance, it ended up being approximately 10% of the variance we found in our empirical Hamilton was accounted for in our oscillations. And we find that the model actually identified specific oscillations as the most important, specifically theta and beta. And interestingly enough, there was a hemispheric sign difference in their contribution to the depression readout. Now, importantly, this model is robust to the inclusion of stimulation artifacts. These are the artifacts caused by mismatch compression over the course of 7 months, and including that as a feature doesn't change the results too much. We found that SCC LFP oscillations could actually be used to predict a part of the depression signal in all of this work. And we're working on putting this stuff out, and the code should be available. The next piece of it is the network action. So to characterize this, we combined LFP and EEG recordings and stimulated at 2 different regions. One on target, which is where the SCC white matter target for each patient is, and then one off target, which is either 1.5 millimeters above or 1.5 millimeters below. What we did is we took recordings before stimulation and during stimulation and found the response, the change during the stimulation from the pre-stimulation baseline, and used these segments. We segmented them into 2-second segments and used these segments to characterize both LFP response and EEG response. And interestingly enough, in the LFP, we saw under the on-target condition, on-target is blue here, we saw no major change in either the left SCC LFP or the right SCC LFP oscillations across all low frequency oscillatory bands. Surprisingly, when we're off target, when we're actually not stimulating the therapeutic target, we see broad increases in activity, specifically in alpha and beta. Now, this is a little bit counterintuitive until we realize that we're actually trying to stimulate white matter tracts. In each patient, we're going to the white matter tracts and we're specifically targeting those. So when we look at the EEG under the similar exact same experiment, so we were collecting EEG at the same time, when we look at the white matter tractography associated with being on target, we see that we mostly hit right cingulum bundle in our patients. And that right cingulum bundle hit is manifest in the EEG by a right-sided alpha increase near the parietal tracts and a temporal decrease. Now, comparing this to off-target stimulation in each patient, we see a very, very different response. These are scaled together. And off-target evokes a very, very different response in the EEG. suggesting we're engaging a very different group of white matter tracks. Actually makes sense. Now, the last thing we did was actually do a classifier on just the EEG segments and found that we were able to achieve approximately a 90% classification accuracy in differentiating between on-target and off-target stimulation using only 2-second segments. And the channels that ended up being the most important, this was using an L1 regularized SVM, we found that the channels that were most important were, as expected with the tractography, this right-sided EEG channels that correspond with the cingulum bundle and left-sided temporal channels that correspond with the left-sided uns fasciculus. And this work is being done in a similar way. I am running out of time, so I'm going to try to, I'll skip through the summary. I think that was fairly clear. We see local changes in the SCC. We only in the long term, not in the short term. In the short term, we see specific network activity in remote regions measured by the EEG. And this gives us a clear idea of what the mechanism of action of SCC white matter DBS is. Now, the modeling work done here is we actually introduced the virtual brain, a dynamical systems platform, a whole brain modeling platform, into the middle of our tractography and our EEG. And what we were able to do is predict where our primary EEG channel changes would be, and then predict where our secondary EEG channels would be by applying the connectivity matrix of the virtual brain parcellations, and then using that as a mask on our empirical EEG measured in each patient. And what we ultimately found was that the primary channels were mostly increases in alpha, and our secondary channels were mostly decreases in alpha. Now, further work needs to be done to validate this in our other patients and at different stimulation voltages, as well as to actually include dynamical neural mass models into the middle using the virtual brain. So in summary, we have DBS of SCC white matter alleviate symptoms of depression and actually evoke specific changes to network level oscillations that we're able to observe at multiple scales for the first time. Oscillations in the SCC LFP can actually be used to partially decode depression state, but obviously other brain regions are involved with depression, so we need to find a way to expand this readout. And careful analysis of LFP data is needed to actually confidently say you're analyzing brain oscillations. Clinical data is noisy and sparse, so actually taking a data-driven approach is challenging. Instead, I would suggest doing a data congruent approach where you take models that clinicians use and ensure that they're consistent with whatever data you're able to collect. And finally, this reverse engineering style approach may be the way we can actually find the long-term mechanism of antidepressant DBS, and tools like the virtual brain and connectivity matrices and connectivity analyses are actually going to be crucial to actually dissect what's happening in our patients. So finally, thank you. Feel free to follow me on Twitter and read a little bit more about the work at my website. Any questions?
Okay, that was great. Yeah, thank you. Thanks a lot. So we have a couple of questions coming in for you. So one question was actually typed in the chat box, so I will just quickly ask that. Are all patients receiving bilateral DBS?
Yes. So when in the experiments I showed today with chronic recordings, yes, chronic therapy is bilateral. And in the acute recordings, the LFP and EEG recordings, they are bilateral stimulation.
Okay. Okay. We have another question coming from Paul such that, Paul, I'm trying to would you like to ask questions yourself? I can make you unmute and you can ask your question yourself.
Sure. Can you hear me?
Yes, we can hear you.
Yeah, very nice talk. Do you see any longitudinal changes in the EEG patterns as a function of whether the stimulation is on target or off target?
It's a great question. So I personally didn't do any of that analysis. Some folks in our group are exactly doing that. We've done the same experiment, the impulse response style experiment with on target and off target at 3 time points at 0 months, 3 months, and 6 months. I don't know what that data looks like. And the challenge there is our EEG is going to be our signal is very small and we're going to be sampling it very sparsely. So I don't know the answer to that.
Thank you.
Thank you. Thanks a lot. Okay, so with that, we are actually running on time. I do have some questions, but probably we can ask towards the end if there's some time. Okay, thanks a lot, Vinit. Again, it was great talk. I really enjoyed it. So let's move cracking. So we have the next speaker, who is Vinicius Carvalho, and he is a PhD student in the Center for Neuroscience at the University Federal de Minas Gerais in Brazil. So Vinicius, it's all yours.
Okay, thank you so much for the introduction, Adil. Can you see the slides okay?
Yes, we can see everything. We can hear you well.
Okay, great. I'm gonna skip the traditional what is epilepsy slides and just highlight that epilepsy is characterized by recurrent seizures. And by predicting or forecasting these events would greatly ease part the burden of patients of epilepsy, whether by enabling the device of alarm of early warning systems that would warn the patient or their family that they have an incoming seizure and they could go to a safe space or something. And it would also allow to close the loop for real-time targeted interventions that could prevent the occurrence of seizures themselves. But this quest for the reliable seizure prediction or seizure forecasting systems has been marked by ups and downs, with some once promising methods that when faced with more real and clinical data and wider data sets didn't achieve as expected. So this has been actually termed as the longer winding road. But in the last few years, with some advances in technology, we have a lot of reasons to believe that we have promising prospects that reliable seizure forecasting systems might be achievable, at least for some seizure types. Among some of these reasons include the availability of long-term recordings and implantable devices, increasing number of public data sets, the use of individualized models of patient data, deep learning algorithms, and the one I'm going to focus on this presentation is the use of active probing paradigms for seizure anticipation. That's basically the idea that you can get more information out of the system by stimulating it and getting its response. So you can get a better picture of its dynamics and the states that the system is in than you would get just by passive observation. And we have some examples of this being used for assessing seizure susceptibility. Around almost 20 years ago, in the first use of this by Stilling Galizing and Lopes da Silva's group with visual simulation. And throughout these years, we have some uses of this strategy with a few other groups, such as Inference and Mark Cooks in Australia, even with patients with epilepsy. Our group has shown some uses of this in a rat PTZ, acute seizure model of pentylenoltetrazole in a rats, that when we use simulation, we can see the temporal rearrangements of some specific waveforms right before the seizure happens, and that's not visible with passive observation. And this is without eliciting seizures themselves, so they have no proictal effect in this model. And also with auditory simulation, with auditory steady state responses in a audiogenic rats strain model. So the objectives of this work is basically combining these two, is to explore different conditions in which this active probing approach is useful for highlighting the underlying parametric changes in a computational neural model that lead up to seizure. And we expect that our hypothesis is that as the model parameters are shifted from normal towards the ictal states, we would highlight excitability and synchrony features with the use of low frequency probing stimulation. For our work, we used the Wendling and colleagues CA1 neuromass model. It's a macroscopic, mesoscopic, or left parameter model akin to the ones pioneered by Lewis and Cohen, Lapsus Silva, and Walter Freeman, in which we have specific populations, neuron populations, and we take the mean activity of each population instead of having detailed models of each neuron. And these we have functions that rule the interactions between these populations, such as the wave triples that represents the post-synaptic potential to actual potential density of each population. And we have the post-wave function, that's the opposite, the actual potential to post-synaptic potential. That's we have the specific impulse response functions for each population. That will be main cells, excitatory cells, that could represent pyramidal ones, and inhibitory interneurons that could be peridendritic, so they have slow dynamics, or parasomatic, so they act really fast. These would represent basket cells. And the inputs of the model is a direct actual potential density inputs, white noise, to the main pyramidal cells. And just by shifting these parameters, these excitatory and slow and fast inhibition gains, we can get a broad range of types of activity that quite resemble the real-time, real-depth EEG signals, ranging from normal background activity to sporadic and sustained discharges of spikes. We're going to focus on normal as we shift toward the ictal-like activity. And we have some other types of oscillations as well. We propose a slight modification to this model by coupling additional population sets through the main excitatory cells. And we rule this with coupling in. And we add the additional inputs that represent the probing stimulation. That will be the low frequency probing stimulation. That can be either to one population or both. And as I mentioned, we can tune the excitatory gain of one or both populations within the one. We can decrease the slow inhibition gain and bring the system closer to seizure thresholds as well. And we can bring seizure data-like states by increasing this coupling gain. And this presentation, I'm gonna focus only on this one. That will be this one model setting that I'm gonna show. We have both populations. We measured the simulated activity of both. And we stimulate only one population while increasing the coupling gain. And we can also simulate both populations simultaneously. Here we have some other simulation parameters. As I mentioned, the probing, active probing approach is low frequency one. So every 2 seconds, we give 10 milliseconds burst. And we vary this with each iteration, with each simulation, ranging from 0, that would be the passive case, the passive observation case, with steps up to 200 pulses per second. That will be the incoming, resembling this inputs. Yep. As I mentioned, we have this gradual parameter shift to bring the population near the ictal threshold by increasing the coupling gain. We have tested the other combinations as well, and we extract features after each simulation. So we have the variance, skewness, kurtosis, the lag-month autocorrelation, and the mutual information between the activities of both populations. There will be a measure of synchronization between them. And we can measure the effectiveness of the probing approach. We can see that there is an incoming seizure, or we can see the underlying parameter change. If these features are correlated with either time, the time of the simulation, or the parameters themselves, the parameter that's shifted. So here we have the results. Again, the first model setting in which we simulate only one population while increasing the coupling gain. And here we have the feature series for various skewness, kurtosis, like one, other correlation, and mutual information for increasing values of stimulation amplitudes. And we can see that for one population, we don't see much of a trend as the seizure is approaching. But for the second population, we can see that for increased stimulation amplitudes, we can see that there are some trends for variance upwards and skewness and kurtosis downwards, and also a bit on the mutual information between them. And when we take a look at the Spearman's rank correlation, these bottom plots here, we kind of confirm these trends that for increased stimulation amplitudes, we have correlation of parameter and the feature. And now another model setting or configuration now stimulates both population sets. They have similar profiles as expected for both populations, and the increased it's way more visible. This change right before a seizure happens, and we also had to make sure that the simulation is not eliciting the seizures themselves. So we measured the onset time of the sustained discharges of spikes for every configuration, and we didn't find any changes. And now concluding, we have that most model settings, the active probing approach indeed highlighted features that indicated these model parameter changes, but we had a local effect. That is, the changes that are not observable in features restricted for the populations that are not perturbed, not approached. And also some feature changes, such as increased like one autocorrelation, variance, and mutual information, are common signatures of phenomena of critical slowing down. This might be related to approaching critical transitions, and it's still unclear if the onset of seizures involves such transitions. We have evidence both in favor and against it. And this might be another use for this active probe approach of highlighting or telling us something about the dynamics of these transitions involved in from normal to ecto activity. And we have our preprint available, so I'm happy to take any questions now or later if you want to get in touch with me. Thank you.
Okay, thank you. Thanks a lot, Vinicius. Very interesting and somewhat related to the work that we are also doing in terms of predicting seizures. Are there any questions for you? If there are any questions, please type them in the Q&A box, or if the panelists wants to have question, they can actually ask live here. So, I mean, this is great. So one thing that I wanted to see on the last slide, you had critical slowing. So there was a recent work from which is coming out of the groups like Mark Cook and others, Levin Kuhlman, which talks about critical slowing as a biomarker.
Yes.
Yeah, so it's so can you, so I was just thinking that is it possible that you can actually replicate or show in your model that you can see critical slowing happening, or certain parameters which can replicate some of the work that they were showing?
We did this with other parameters. These are included in the preprint, but we saw these changes as well, that decreased like one autocorrelation, the variance, which might indicate that the response to the stimulation is getting more and more slower as the transition from normal to ecto activity is approaching. So for this model, we see that we have some evidence that this might be indeed the case. It will be interesting to test other models. I'm curious about models of absence seizure, for instance, if this will be the case. And
yeah.
And
Okay,
of course,
good.
test this with real data, with animal models that we're in plans of doing this also.
So I mean, so I was just thinking what the main idea here is this, that if you can have in silico seizures, so if you understand how to generate seizures, then you would be able to, because then you know that how the dynamics changes in the pre-ictal state to the ictal state, and then you can use that insight and those characteristics, some of the features, which can then be used to do prediction of seizures beforehand with a certain, seizure horizon, or a prediction horizon, that you can develop a warning system. So that's basically where you are going, right?
Yes, exactly.
Okay.
And the model that Vinit mentions, the peripheral brain, that's one goal that we have as well. We tested a similar approach on that epileptor model. That's from the same group, and the results are quite similar. But I really wanted to take this and put it on test it on the peripheral red brain.
Okay, thank you. Thanks a lot, Vinicius. While we have Vinit here, I wanted to just quickly have a question for you, Vinit. We have a minute or two before we can close. You are still on mute. So if you would like to unmute yourself. On your, in some of the last one or two slides, you were talking about virtual brain and using neural mass model, because the idea is this, the whole thing is this, that we actually don't know the mechanisms, underlying mechanisms, that how the deep brain stimulation actually works. I mean, it works, and we know that it works, and we don't know it works for some, it works for others, and we actually don't know. And so understanding the local, what are the underlying mechanisms are the probably the holy grail. So what was your thoughts about how you could use this in terms of understanding mechanisms using virtual brain?
Yeah, no, I want to say it's very relevant. I really like the approach you're taking, Vinicius. This idea of active probing, of impulse response, I think is crucially important, because that's kind of what we were trying to do with the on-target, off-target. Is can you put the same signal in one region and a different region, and do your best to identify where it propagates, how it propagates. So there was one slide that I wasn't able to include for time reasons, but from the virtual brain connectivity, I'm trying to take an approach from nonlinear controls. So actually leveraging some of the really beautiful work coming out of Seattle, groups like Brunton, doing autoencoders to learn dynamics directly from time series, multi-dimensional time series, finding a low dimensional representation that you then decompose into its nonlinear basis functions. So I see TVB as a fantastic bridge from empirical observations of EEG and LFP into biophysically constrained dynamical systems models, and then the use of these more modern tools to come in and shape around these idealized neural mass models that we've kind of found the average of. Does that kind of answer the question? I think
yeah, I mean, I think there's a lot of parallels between your work and Vinicius. So I think when you were talking about the work with where you can actually use VAEs to learn, the problem with the epilepsy work is this, that there's not enough data. So the problem is the data, because in order to, for example, Mark Cook has this beautiful data set, but it's from three patients, right? Or the other data sets. So there is not enough data to actually train So we have been doing some of the stuff where we are actually using generative adversarial network GANs to actually augment, to simulate data which looks like the actual data, and then use that.
What I would highly, sorry, I just want to make sure I tell you about this tool called Cindy, SINDY.
Okay.
Are you familiar with Cindy?
Not really.
So this is out of the Brunton group. I'm just getting familiar with it, but the entire premise of it is sparse identification of dynamical networks. It's S is sparse in Cindy. So
okay.
There are people working precisely from more of the Bayesian perspective to say, yeah, you only have 3 patients, but whatever structure you see in those 3 patients is probably going to be very conserved. So find me the most conserved thing. Otherwise, I would agree. The data it's sparse with respect to the variables that we could possibly have, but there are tools that people are developing that can make the most of whatever data we have.
Yeah, I mean, this is exciting area. There's lots of things happening recently. So yeah, thank you. I think we can go on and on with this, but we have to stop here. It was really good to have this conversation and to know your research. And I definitely will be looking at your work. Okay, so thank you. Thanks a lot again. And from here, thanks everyone, and goodbye.
Thank you so much.
Okay. Thank you so much.
Bye-bye.
Bye-bye.
Gagana, perhaps it's good to mute yourself for right now, and then when Jonathan's done, great. One minute to showtime.
Less than a minute. Now.
Okay. Hello all. Welcome to this session on the computational modeling and techniques. My name is Kirti. I'm an assistant professor at the University of Tennessee in Knoxville. My research interests are mouse models for neurodevelopmental disorders with a focus on cellular and behavioral neuroscience. I'm really excited to be your MC today, and along with Dimitra, who's our backend host, we are looking forward to learning more about your thoughts and approaches to these very long-standing neuroscience questions. Please ask questions in the Q&A and post chats to stimulate relevant discussions. Since we have to abide by the time restrictions, I would like to request the speakers to please keep to your allotted time, 12 minutes plus 2 minutes or so for questions. I'll hold up the signs for you for time. So first up, I would like to introduce Jonathan Cannon. He is a postdoctoral associate studying prediction of autism spectrum disorders in the Sinha lab at MIT. He's also working with Anirudh Patel on neural models of beat-based timing. Outside of his academic pursuits, he plays rhythm on violin with Boston klezmer band Ezekiel's Wheels. He'll be discussing a Bayesian filtering framework for neural and sensory motor entrainment. Jonathan, please go ahead. Is there a problem with the screen share, Jonathan? We can't hear you.
Unmute. Okay, sorry about that. There's my screen share. There we go. Okay. Sorry about the delay there. All right. Hi, thank you so much for coming. This has been some modeling and computational work that I've been doing over the pandemic, and I'm really excited to get any feedback on it that you're willing to share. So as my jumping off point, I'm asking the question, what is optimized by entrainment, both neural and sensory motor? And here I'm defining entrainment as the temporal alignment of a biological or behavioral process with irregularities in an exogenously occurring stimulus. It's a pretty general definition. A common example is tapping along with a metronome. And in that, a common way of understanding that behavior is that I'm minimizing timing error between my taps and the clicks of a metronome. But thinking about it this way generalizes poorly, because you can entrain continuous movements to periodic sounds. You can tap on the offbeats of a metronome and not try to align the sound in your taps. All of those are entrainment type behaviors. And so a way that I found really useful to think about this is as having an intermediary step where we're continuously estimating the phase of an underlying cycle. And then that estimate is now informing our behavior, which we can optimize in different ways based on that estimate. This allows us flexibility of our behavioral goals in entrainment. So formalizing it like that, we actually end up with a pretty nice mathematical statement of what it is we might be doing in entrainment. So if we suppose that a stimulus is based on some hidden phase, phi, that advances sort of like a clock, or along the real line, but advances steadily underlying the stimulus, then we can continuously infer a distribution on that phase based on noisy sensory measurements using Bayesian methods. And that inference effectively amounts to that intermediary step where we're doing that phase estimate. So if we make the variational Bayesian assumption that we're only keeping track of a mean and a variance of that distribution, then our best estimate at any given time for the phase of this underlying this stimulus is a normal distribution with some mean mu that entrains to the hidden phase and a measure of uncertainty or variance C. So then the question is, what type of data are we using to inform phase inference? Inference of hidden state is often modeled using predictive coding type models in the brain, in which the sensory stream is modeled as a continuously fluctuating values over time. And hidden state inference happens through minimizing the errors between predictions of those sensory values and the actual values. But in entrainment, we're usually being informed by the timing of events, especially auditory timing of events. Error is the error we're minimizing is timing error. And predictive coding models as they currently exist aren't actually well equipped to do this over continuous time. And so the formalization that I've come up with here is to assume that events in the sensory stream are generated according to a point process. And that point process has a rate function that is a sum of Gaussian shape functions with different parameters. And so you can have more or less precise expectations over phase. And this function I'm referring to as a temporal expectation template because it refers to the times that we expect events to happen as a function of phase. And we've got these, each one has a mean and a variance and a scale lambda. And then there's also this additional constant term, which says that events can happen that are not associated with a specific expectation at a certain rate. And that's the height of this function at baseline here. And by using variational Bayes methods, in particular point process Kalman-Bussy filtering in continuous time, it's a math that exists, but you kind of have to dig for it, we can invert this generative model and use the events to continuously estimate phase and a mean variance on the posterior distribution on phase. So what does this look like in practice? Well, here we've got a graph that has on the x-axis the phase at which an event occurs, and on the y-axis the updated phase estimate after that event occurs. And so these little plateaus here show that if an event occurs close to an expected event time, then the phase is corrected to be even closer to the mean of that time. But in between, you can see that if an event occurs in between expectations, then this diagonal line says that nothing happens to the phase. And that's actually because that event would be attributed to that background rate of events occurring that are not attached to a particular expectation. And so this phase adjustment gives us the ability to entrain our phase estimate to the underlying cycle. Here you can see the dynamics of uncertainty, where if an event occurs right at the expected time point, then the uncertainty or variance decreases. But if it occurs close to that, it actually increases, signifying an increase in the uncertainty about your entrained phase. And then if it occurs in between, nothing happens. And here we can see actual entrainment to a real heard rhythm. So I'm putting in a temporal expectation template that happens to be a swingy set of eighth notes, and those emphasized or accented eighth notes are expected with a small variance and a strong expectation. And then the offbeats have larger variance and or weaker expectations. And as you hear a rhythm, in this case, da-da-dum, ba-dum, ba-dum, each of those events is incorporated into a phase estimate. And you can see through the gray shading here that the uncertainty about the phase estimate decreases at each event. And the line shows that phase is tracked through the whole rhythm. And if they're on the right, we can see if there's a phase shift. In other words, all of the timing of a whole set of events is moved up a little bit. That phase shift is quickly accounted for by this model. And there's an increase in uncertainty, but then it catches back up and figures out where it is. And I'm calling this PIPIT, by the way, I forgot to mention. It's a phase inference through point process event timing. So moving on from there, one fun effective PIPIT is that it fails in a similar way to the way that humans fail. So in this case, I'm giving it a very difficult rhythm, da-dum, bump, bump, ba-bum. And when I do that, a lot of these events coincide with weak expectations. And during that long silence, uncertainty increases and events get misattributed to strong expectation points until at the end, it's just completely bollocksed up. This is very similar to the way that humans fail to track excessively syncopated rhythms. Another really interesting effect of this particular framing of the problem is that it actually predicts that in between events, expectations can warp the progression of estimated phase. And so here you can see that if expectations are very strong, then and expected events are omitted, then that distorts the passage of phase. And when an event actually occurs at the end, it seems like it has happened earlier than expected. And there's actually evidence of this illusion in human perceptual report and tapping data that's been reported multiple times since 1886. And finally, we can actually do inference on both phase and tempo in this way. So if we just give it a forward model that has phase advancing at a variable rate, theta, and theta has some noisy drift, then we can do PTIPIT, phase and tempo inference from point process event timing. And PTIPIT actually accounts for some other interesting human data showing that if you're tapping along with a metronome and the inter-onset interval is very large, then timing perturbations will be overcorrected for those very slow tempos. And PTIPIT will do that because for long inter-onset intervals, there's more accumulation of uncertainty, leading to larger corrections. And when tempo is corrected in addition to phase, then you end up overshooting the mark. So big question for a neuroscience conference is this neurally plausible? So I've been working with Ani on a working hypothesis for how the beat is tracked in the brain. And our idea is that the supplementary motor area, the neural firing state and supplementary motor area keeps track of the phase of this external stimulus, that the basal ganglia is involved in laying out immediate expectations in the immediate future and sort of setting up that template for you right into the future. That expectations are then projected down to auditory cortex from motor areas via the dorsal auditory pathway. And that event timing errors based on those expectations are then relayed up the dorsal auditory stream. That process may involve cerebellum. We're still talking about it, not sure based on experimental data. And finally, an important part of this whole system is that certainty or variance of these phase estimates is tracked over time. And there's reason to believe that striatal dopamine plays the role of an index of temporal certainty. So we've got a preprint on that, which I'm linking to here, and I can put into the chat after I'm finished. I want to thank Tom Kaplan and Ani Patel, Nori Jacobi, Ed Large for very helpful conversations, and Carl Friston for one very helpful email. I think that's all I've got to share. I'd love to hear your thoughts and questions.
Thank you, Jonathan. That was really exciting and interesting, and you're ahead of time, which is fantastic.
Great, great. The nervousness plus practice turns into rushing.
Right, yes. So we have a few questions coming in. So I'm going to, they're not in the question answer, so I'm going to read it to you off of the chat system right now.
Yep.
So one question is, how does your model calculate frequency? Does this calculation suggest or reject possible neural substrates?
Yeah, so the question of, I mean, I guess by frequency, we're talking about the period of the beat or the tempo.
Yeah.
And for that, it starts with a prior, it starts with some distribution over tempo. And then based on that, it's doing this variational inference on both phase and tempo. And so when an event occurs early, then both the phase and the tempo are updated to a posterior distribution based on the fact that that thing has happened. And so it's all done through this variational Bayesian machinery. That leaves out the results that suggest that tempo updating may need to be conscious. And so it's sort of a first approximation here. In terms of neural substrates, the way we're thinking about it is that that's got some persistent representation in prefrontal cortex, dorsolateral, possibly prefrontal cortex. But yeah, our thinking about the neural substrates of tempo inference is not as developed as phase inference.
Right, great, thank you. The next question is the point process a convenient assumption for formal purposes, or is it really worth throwing out a lot of continuous process?
So, so formally, I mean, it's very useful. I feel like it's the best mathematical tool we've got to talk about timing. This model can quickly be extended to include continuous input as well. That math also exists, and I'm just leaving it out for simplicity. And so you can take both continuous and timed data and integrate it into an inference about phase. So you don't need to throw out continuous data entirely. I mean, I think that the assumption that events are generated as a point process, I mean, we also end up with a possible generality of introducing history dependence, which again, I leave out for simplicity, but this would lend itself to that. And once you've got a point process with history dependence, that's really as much generality as you could possibly need to describe event timing data as it comes in. So I think that it's a mathematical structure that really doesn't limit you much at all once you are willing to bring in history dependence and optional continuous data.
Thank you. I have questions for you, but I think I will, in interest of time, I'm going to wait a little bit longer. And thank you again for a wonderful talk and starting us off.
Thank you guys for coming.
And if you could stop sharing your screen, we can get Gagana back in here. So in the meantime, I'm going to introduce her. She is currently the president of a nonprofit open cancer organization, AI Without Borders. She was previously a research fellow for initiatives at the Stanford University. She recently presented her work on multimodal interactions of schizophrenia in ICML 2020 and will be talking about her work on neural pathway establishment at NeurIPS 2020. Today, her title is Being in Sync: How Neural Entrainment Affects Attention and Conscious Experience. Welcome, Gagana.
Thank you. Thanks, Jonathan, for a great talk. And I'm Gagana. I'm going to be talking about how neural entrainment affects attention and conscious experience. My co-authors, Deepak and Mehdi, are here today, and they will parallelly try to answer your questions on chat, and we will take your questions at the end as well. We would like to thank Kyle and Shaul for being our mentors on this work. So I'm going to kickstart with a discussion on the types of brainwaves, and then discuss a little bit about our observations on synchronization across different brain regions, and finally explore how synchronization mediates conscious experience. Brain waves or neural oscillations are rhythmic patterns of neural activity, which neurons use to communicate across different brain regions to communicate emotions, thoughts, behavior, And this ranges from low frequency delta, which occurs in deep sleep, to high frequency gamma, which occurs during problem solving and cognitive processing tasks. In the past, we've seen that synchronization sort of affects cognition, working visual memory, rapid learning, and also interpersonal interactions. But interestingly, synchronization itself is also speculated to lead to conscious awareness. There is no direct evidence, but this question has been explored experimentally multiple times over the years. And in this work, we try to establish direct evidence through computational means. So when we talk about synchronization across brain regions, which is represented here from LFP data of rodent's brain, where the connections here, the color and the thickness of the lines here represent the strength of the connection. We see that the visual cortex does not have a very strong correlation. So that is probably because in a rodent's brain, only 1/3 of the visual inputs get processed because of the lateral position of the eyes. And the second important observation we made is that when we look at the correlational data across regions and within regions, we always find that there is a pretty strong correlation within regions, except for the hippocampus. The hippocampus is always a clear outlier. And this is probably because of the complex asymmetrical architecture of the hippocampus, which has different neuronal types with distinct spatial firing patterns. And that has not been explored over time, and how they integrate into a coherently functioning metanetwork in the hippocampus. The hippocampus also, or the sharp waves and ripples also originate in the hippocampus. And this probably leads to interferences with trisynaptic loops. So the synchrony is not well captured with respect to that. So what we observed across correct and incorrect trials is that there is always a distinct, consistent communication pattern over correct trials, and over incorrect trials, it is more inconsistent. So this shows that there is some consistent communication happening over correct trials as opposed to over incorrect trials. So we try to capture this consistency across trials with respect to synchronization in a new attribute that we propose called the synchronization consistency factor. Now, when we employ theories of compositionality and componentiality of consciousness across brain states, the mice is given a visual cue to which it's expected to pay attention, followed by which it will take some kind of action that is interacting with the environment, and for which it will receive some kind of a reward for doing the task correctly or as expected. Over this process, we realize when we look at the phase synchrony across attention, action, and reward, we realize that there are multiple observations that we can make over this process. So the first one is that when we look at the midbrain basal ganglia interaction over time, we realize that this increases gradually, especially in the reward phase. This is probably because the dopaminergic neurons in the midbrain constantly communicate with the basal ganglia, and this establishes the reward. This gives us the reward phase, reward part of the phase synchrony. And when we look at the second observation we can make from here is that the hippocampus also consolidates the learning part, which is captured over time in the phase synchrony, and that sort of consolidates into the working memory. So that mediates the experience of consciousness, where higher order embeddings are captured for learning and cognition over time, which manifests as intelligence in the rodent. So while we have explored phase synchrony and correlation over multiple networks and across brain regions, the intelligence and the consciousness in terms of individualistic neurons is a question that is yet to be explored. We would like to thank our other contributing team members, Kirtana, Manil, Sitari, Abida, and Nilofer, for their contributions in this work. We would also like to thank the organizers of Neuromatch and Neuromatch Academy, as well as Nick Steinmetz for the data. And we are now ready to take any questions that you might have. Thank you.
Thank you, Gagana. Wow, you guys are all like ahead of time. This is fantastic, which means we could have more discussions. So that's really great. I'm going to start by one question that's in the Q&A. The consistency factor of synchrony over trials. What does that mean? Trials of what and consistency between what?
We have multiple mice, which is around 10 or 20, and we have multiple trials. So the mice are expected to perform the same task in different ways, and we have then split it into active and passive trials, which means that the mice is allowed to interact with the environment in some cases, and the mice is not allowed to interact across the environment in some cases. So when we just look at correlation or synchrony, these terms don't capture how they behave across trials. So we included a new factor to incorporate how the mice behaves across different trials. So that is the consistency factor that I was talking about.
Okay. So just to follow up on that, so it's mainly talking about different trials of the animal, the same animal, but across different trials that it's performing this.
Yes.
Is that correct? Yes. Okay. All right. Any other questions that are coming up by any chance? Okay. I have a question for both of you, which is this idea of learning in this component, right? So for anything that's related to behavior, or from Jonathan's talk about learning music or learning rhythm in music, are you guys talking mostly about the adult animals and already well done trials, and how does learning play a component in this aspect? Perhaps Gagana can start, and maybe Jonathan can pitch in a little bit. Yeah.
Yes, we are talking about adult animals when we talk about the learning processes. What we speculate is that when the mice performs, like I specified in my talk, there are three aspects of the mice's behavior. There is the attention phase, the action phase, and the reward phase. So when the animal repeats the same task over time, then it sort of tries to learn that, okay, I am getting a reward when I perform this task a certain way. And over time, it sort of understands this pattern and tries to perform in a similar way, so as to get the reward in the short term, and the learning happens over the long term.
Jonathan?
Yeah, thanks. I think that's a good answer. In my case, I mean, I think learning has to happen, learning of temporal structures in particular has to happen through some kind of bootstrapping process, because these temporal expectations are attached to this underlying phase structure. And so you need to have some inference about where you are in a structured stimulus before you know where to attach your temporal expectations. But then you need those expectations to inform where you are in the stimulus. So yeah, it's a tricky problem how we build that up. But I've been looking at some models for learning from, I mean, in the case of Kalman type filtering like this, learning while filtering, while learning while filtering. And if there are ways to avoid the loops and awkwardnesses that lead to. Friston and company in 2015 wrote something about that. And so I'm interested to start trying to incorporate those questions.
Great, thank you. We have one more question from Nadine. So this is for Gagana. You're talking about conscious experience. Not sure whether we can tell something about conscious experience in mice. What's your definition of conscious experience? Great question.
So when we, so consciousness in itself is a very broad field. So in our case, we try to sort of focus it on attention, action, and reward. and sort of try to minimize this broad definition into more simpler problems that we can answer. But conscious experience in mice is still an ongoing research problem, and is still there are multiple definitions of consciousness and multiple theories around consciousness, and we can sort of break down this problem in different ways. We looked at, for example, if you look at Marvin Minsky's idea of consciousness, he splits it into 21 different parts. So there are multiple definitions of consciousness. So for the purpose of this talk and considering the limitations of time, we just decided to break it down into 3 parts. And consciousness in mice is still very broad area.
Yeah.
So I hope that answers your question.
Yeah, I can completely understand. The reason I'm excited about it is because we study mice behavior. And so even trying to separate them out into movements versus actual volition, right? Motivation towards a behavior versus actual movement of a behavior versus what is the mouse actually thinking when it's doing this. It's a hard problem to solve and to get into. So great questions, great discussions. Please have them coming. And of course you can post them on the Q&A and continue this discussion forward as well. Thank you, Gagana. And we are going to move on to our next speaker, who is Rob Law. He is currently at the Brigham and Women's Hospital, Harvard Medical School in Boston. He's working with Sean Purcell. His title is Data-Driven Discovery of Dynamical Relationships within Electroencephalographic Spectra in Sleep. Welcome, Rob.
All right, thank you. And thank you to Gagna and John. Those talks were much more elegant than mine is going to be. The data that we're working with is kind of a mess. And this is the object of study here. It's a spectrogram from one channel of sleep EEG over an entire night. And our motivations here are epidemiology and population genetics to some extent. What we're really wondering is how we might collapse several hours of sleep EEG into an endophenotype. And to presumably get toward the question of what aspects of longer time scale sleep dynamics are trait-like or disease biomarkers. This is very much work in progress. So typically sleep is staged where each 30-second epoch is assigned a category to non-REM, 1, 2, or 3, REM or wake. And often these spectra are averaged over, say, like all non-REM epochs to characterize sleep for an individual. But these spectra do vary slowly over time, even within a non-REM or REM episode or a sleep cycle. And we want to be able to quantify these things. And so in what we want to be able to do is kind of compactly describe how activities in different frequency bands interact or evolve over time. So this is a little bit of background here. So power in a given frequency band can usually be thought of kind of as a proxy for a density of events. So this is a sleep spindle here, and it turns out that on average, power in the sigma band corresponds with density of these spindles. And there are a number of these different kind of types of events: slow oscillations, delta rhythms, sleep spindles, beta events. And thalamic activity appears to be a common source for a lot of them. Which brings us to a model that kind of motivates us here. And this model poses that thalamic neurons can essentially be either in beta, sigma, or delta modes. And as sleep deepens, you go from beta to sigma to delta. As sleep lightens, you go from delta, sigma to beta. And they modeled this as a linear system of ODEs, or actually it was a Markov process, which is kind of the same thing, but a little bit more restricted. What I think is a key insight here is that a dynamical systems model can describe how frequency bands interact. And so what we're going to do is try to fit ordinary differential equations to these data and the data that we have, and use these results as sort of a guide. But it's well known that individual spectral modes vary. So delta for you might not be the same as delta for me. And this is best studied in alpha, where you can see and this is a study from Kathy Kerr, that in individual spectral peaks can vary from 7 to 13 hertz. Possibly more. So the question is, can we characterize the evolution of band power in a population when bands are not even the same necessarily across individuals? And so what our approach is to characterize how these band limited activities evolve relative to each other. We're going to use non-negative matrix factorization to factor the spectrograms into frequency and time series factors, and that will give us a handful of time series that we can fit linear ODE systems to, okay? And so this slide I'm not going to be able to explain super well in the time that I have, but what we actually do here is 3 rounds of non-negative matrix factorization, first on the individual level, second on the group level, and then third again on the individual level to get frequency and temporal factors. But after this whole like kind of reseeding process, these spectral factors are very consistent across individuals. I mean, almost like by design, but they also admit some variability. And without having kind of presupposed this, the 4-factor model yields delta, sigma, like a slow beta and beta gamma component. And while 6 factors adds like a few more components as well. All right, so we've used NMF to reduce the frequency axis essentially, and now what about the time axis? So we're going to use these ODEs to reduce this dimensionality, or attempt to anyway. Just as an aside, the mean dynamics of sleep onset in the chat data set that we're using recapitulate some aspects of like the key aspects of the America Fortune data that use the classical delta, beta, and sigma frequency bands. Although ours is log transformed, theirs is not. All right, so what we're going to do first is we're going to smooth the data. We'll fit the dynamical system using the Cinder package in R. And essentially what we're doing is regressing X onto dx dt. And the kinds of questions that we can get to are whether sigma power, for instance, correlates with a change in delta power, and rather than, just does it correlate with delta power itself. So this is what we wind up doing. This is what this kind of looks like. You have a smooth time series. The actual time series is below. It's all rescaled. And here, let's see, I don't really have a pointer, do I? Okay, but anyway, in the bottom left corner is the smooth time derivatives, whereas the modeled time derivative is in the bottom right corner. But what's kind of key to notice is that sigma and d delta dt are pretty well correlated in this case, and it turns out in many cases, like in this entire data set, the coefficient for d delta dt versus sigma is consistently positive across this population of about 400. So this data set yields a consistently positive correlation between delta slope and sigma power, and this is consistent, I think, with the America Fortune work, and they also pointed to a negative correlation between beta slope and sigma power, which we also do observe. Okay, so, and this is maybe not that important, but like we can look at cases where the d delta d sigma is negative, and these seem to be like kind of aberrant cases that are where things just are kind of decoupled in a way that we wouldn't have expected given like most of the population data, and there is still some overfitting here, so still trying to work out some of those bugs. But But in summary, we combined non-negative matrix factorization and ODE fitting to investigate the spectral dynamics. And this is a flexible data-driven way to get some of these band-limited activities. And we found some consistent low-dimensional descriptors similar to those that were previously modeled. Although it's worth mentioning, we didn't find much evidence for any trait likeness in these coefficients. And so, yeah, the next steps for us, I think, are to regularize the coefficients, apply these to some other segments that haven't been studied before, the dynamics have not been studied before, and I think it's possible to apply this at some finer resolution to maybe look at individual events and to maybe segment out events that we didn't know existed before. Hopefully that would be really cool if that worked. And it's possible that this stuff can be applied to chronic recordings during wake. And the wishful thinking level is that for using this sort of thing for cross-species comparisons. And since you guys are the very first people to see this, thank you. That's about it. That's about all I have for that. So all right. That's all. Thanks.
Thank you. Those are beautiful graphs, I have to say. While we wait for questions, perhaps I could ask one. You were mentioning that there is individual variability across different people. I was curious about just the same person, but a different day, right? So when you are in looking at your sleep patterns.
Yeah, so some of this so far, we haven't really been able to find any consistency in the time factors. Presumably in the spectral factors, those should be relatively consistent because they're known to be consistent for like using other sorts of other methods, so.
Okay, all right, okay. You also mentioned the sleep component of the sleep in the kids component earlier. So I was wondering, do you have data in tracking babies or kids that go through sleep training? Like, do you see these kind of differences? Is this I would expect that those kind of data are probably pretty noisy. And so I was curious how your program would work for those things.
Oh, yeah, I mean, the noise is an issue. I mean, like a lot of these things are, I mean, all of these data have been run through by an expert. Like we just happen to have a large collection of that data. So it's like they have, for instance, like, basically cut out segments of the EEG that, just, are super noisy. However, this does seem to work pretty well for pulling out wake periods. For whatever reason, the noise spectrum that is involved with all the EMG and everything seems to be like taken care of reasonably well by this gamma component.
Mm-hmm. Okay, very cool. So we have questions coming in. So one question is, do you think the delta slope spindle power reflections, slow waves, and spindles coupling?
Yes, but what I think may or may not be going on is that there's some feedback process between the cortex and the thalamus that's that is using spindles to change delta power essentially. I mean, it's like it's possible there's something causal going on here. I don't know for sure.
Okay, right, okay. Another question is, can this method be applied to other EEG data, not sleep data? That is, what is the length of data required to do a similar analysis?
This I'm not exactly sure. We just happen to be able to, I mean, the segments that we're using are 30 seconds long. However, I'm hoping that this sort of stuff can be, applied at like maybe the one second level or so. I mean, it depends on, I guess, the phenomena of interest, right? And your sampling rate and like that kind of thing.
Great. Looks like there aren't any other questions coming in at this point. I do have one more question since you were mentioning the cross-species comparison. There's a lot of EEG data in other rodent models, right? So have you tried to see if this would fit in? I mean, the sleep patterns are different, but do you think it works out this way?
Oh, I mean, I told you this is wishful thinking. I mean,
yeah.
But I mean, like what I do know is that at least from what I've read anyway, in rodents, the alpha frequency is different, right? So, I mean, being able to kind of align these comparisons, like align the components would be, but then we'd have to kind of check to see whether the dynamics were similar in any way, and these kinds of things. It's like these homologies would not be easy to find regardless.
No, not at all. I don't envy the job, but I was just curious. So let me see. I think that's all the questions we have. Do the panelists have questions for each other? We have a few minutes. So if you guys want to talk about anything in particular about each other's work, you're welcome to do that. I find all of this absolutely fascinating. It's really cool that there are these models that exist for looking at data at this level. Talking about Gagana's question, and there was some aspect of this, which is the higher, bigger question, right, in neuroscience in general, right? How does the brain contribute to the behavior? And you can think about vice versa. How does the behavior contribute to the brain? And you can think about consciousness and intelligence and sleep and all these meta-related subjects. So Jonathan, yeah, feel free to unmute and yes.
Yeah, Rob. So one question that was on my mind was, I mean, I know that a lot of these rhythms have been modeled at like neuron-specific levels of detail with different types of channels and all of those things. How do you see the relationship between your approach and those perspectives on those rhythms?
So right now, I think that's it's has to be vague right now. I mean, but what we do kind of know are that, things like spindles are not just assigned to, I mean, it's not just ion channels. It's really like kind of, there's a recurrent process essentially that's happening. And like my hope is that these processes are linked in this sort of, at the level of something that might be able to be described as a dynamical system. But yeah, but that said, I think part of dividing these things into different frequency bands is essentially like we're trying to, like I feel like the frequency bands themselves are more indicative of often indicative anyway, of time scale than they are of necessarily like rhythmicity. Yeah.
Yeah, that makes sense to me. So you're sort of breaking it up and saying like, all right, this time scale is, it's got some set of ion channels that may be related to it, but we can lump that all together and just say, all right, let's model that with a higher level dynamical system, and then hope that, we can lump together the things associated with this time scale.
Mm-hmm. Yeah. And I would hope that these sort of these factors might be associated with, like multiple timescales on like a single process. But that would be, I mean, again, kind of wishful thinking. I do have a question for you too. So where is the circle, right? So like
I'm trying to write that right now into this paper. So I think that one strength of this framework is that it can talk about aperiodic temporal expectations too. It doesn't need a circle. It stays as on the real line. And you can just pose, to talk about periodic expectations, you can just make a periodic temporal expectation template. On the other hand, that leads to it's sort of blind to the ways in which periodic expectations can be different behaviorally and neurally. And it also leads to some weirdness, like if your confidence in your entrainment is low, then the system needs to know whether you're on beat 5 or beat 6 before it can resolve uncertainty. So yeah, I mean, ultimately the reason I didn't end up putting phase on a circle was because it did almost everything that I wanted it to without doing that, and because the math was harder. But that's a good question.
We have one more question. I believe this might be for Rob. So do you think we can compare non-human primates and humans with this approach? NHP might have more similarity to human data than the rodents, maybe.
I mean, hopefully.
Right.
I mean, this is really in its infancy. I mean, that said though, non-human primates might be difficult because we're actually maybe not. I mean, if we have many nights from a non-human primate, that would be one thing. But typically we need like a large population to kind of to condense some of these factors. Yeah. So I have one question for Gagna. the phase coherence. This is across like all of the regions that you're studying. It's like a global coherence, like a Hilbert phase coherence. Is that what that is, or is that some other kind of coherence that I don't know about?
We're talking more about time-lagged cross-correlation metric between different LFP signals across different regions.
Okay.
I have one last question before we finish up, and this is for Jonathan. I was curious, did you get into these kind of calculations because you're a musician, or did you get into this because you're a neuroscientist interested in music?
I planted that in my intro. I mean, that does sound actually like sort of the same thing. It was my interest in music that led me down this particular rabbit hole, and because I couldn't play live music with friends during the pandemic, this was like the best alternative. I think that was the real origin.
Very cool. Well, thank you all. This was fun, and I learned a lot. This is exciting work. I hope you will continue to the discussions on Twitter. We have your accounts posted, and of course, continue keeping online and chat with each other. And this was a great session. I enjoyed a lot. So thank you very much for coming, and thank you to the audience for wonderful questions. This was a lively session today. So thank you all, and we will see you later. Thank you, Dimitri.
Obviously, it's now stopped because of COVID, but I hope they will, continue doing that. And there is a longitudinal follow-up on the imaging participants as well. So there's already, I think, 3,000 participants who have data on two different imaging data on two different time points. So it's a huge amount of data
that is amazing, because you can see the effect on the long term, that is the causal effect or not.
Are they absolutely?
You said that they are paused right now. I see a question by Pedro. Pedro, also, if you want to go on the screen, feel free to ask me. Congratulations for your work. In the future, are you interested in dissect which of the metabolic syndrome components have greater influence on white matter atrophy or cognition decline? Or do you think that those alternations as a whole are equally implicated?
Yeah, thanks for this question. So from the analysis that we did, we can already look at which factors have the strongest association with white matter atrophy, and this was the strongest factor was obviously hypertension, and this is very consistent with the previous literature. So hypertension is the highest risk factor for white matter hyperintensities in this case. In terms of cognitive decline, I don't think we looked directly at how metabolic syndrome is related to cognition in a direct way, but that is certainly something that we could have a look at. I think also because metabolic syndrome is sort of a cluster of symptoms, so it consists of all those, so hypertension, diabetes, dyslipidemia, and inflammation. I think it's also useful to look at it as a whole, especially given that most of those symptoms actually co-occur, right? So you have obesity co-occurs very often with type 2 diabetes, hypertension, and obviously dyslipidemia. I think it's important to look at it as a whole, but also the factors that are like the single factor that is most significantly associated with white matter atrophy is definitely hypertension.
Thank you so much. Thank you so much, Philip. That was great talk. I thank you again, and I will move on to the next speaker.
Thank you.
So in textbook, synapses are usually modeled as like 101, meaning that like one neuron is connecting another neuron in just one synapses. However, we know that this view is very simplified. One of the cell types that doesn't fall into this simple picture is astrocytes, that they are usually like connecting a bunch of other neurons and have very weird morphologies. To understand the rule and the function, we need to model their network. Today, Marco is presenting a mathematical framework for Neuron-glial network. Marco Pina Garcia is a post-bachelor researcher associate from San Marcos University, working with Dr. Walter Cabrera. And today he's going to talk about mathematical representation of neuron-glial networks. Thank you again, Marco, for joining us today. And we are ready to hear your interesting talk.
Okay, I will share my screen.
Would be great.
Thanks for accept my abstract. I will expose a mathematical framework as is in the title, mathematical representation of neuroglia networks. Well, the brain has more than neurons. The brain has astrocytes, and we know that astrocytes are also implying cognitive computations. Well, as a cartoon of this complex network, I have some diagrams. Well, the basis of neuroglia networks is that tripartite synapses that imply that a neuron interact with another neuron, but also with astrocytes. Neurons synthesize neurotransmitter, release neurotransmitter, and are received by the other neuron. But also astrocytes have neuroreceptors, and astrocytes also have a response to this neurotransmitter, increasing the calcium levels, the cytoplasmic calcium levels, and there are some waves of calcium that travel along one astrocyte to another astrocyte, and that's the astrocyte network. Astrocyte has have networks also. These networks not have synapses in a classical sense, but have gap junctions, and the gap junctions are quite similar to electrical synapses because it allows some signals to travel along two cells, IP3 calcium, but also astrocytes release neurotransmitters, release ATP, glutamate, and can activate neurons also. So it's a complex network with interaction from one side to the another side and vice versa. So if you want more information about astrocyte networks, neuroglia network, you can read the book of Theta, more information about computational glial science. Now, mathematically, I will be not too formal because I know this is not a congress of mathematicians. I will be more informal. So we have astrocytes and neurons, but to model the network as a whole, we need a term for both, and the term is a suggestion is general neuron, I think, because astrocyte also synthesize neurotransmitter, is affected and receive neurotransmitters, so it's like behaving like a neuron. So it's a general neuron. Well, and we need a set for this general neuron. We call the set X, the GN set. And we have that this set is the union of the set of neurons and the set of astrocytes. And now we have to formalize the interaction, the complex interaction between astrocyte neurons or neurons and neurons or astrocyte and astrocytes. And we need a general framework for that. So we can see synapses as the graph of a binary relation. What's a binary relation? A binary relation is an ordered tripole that has a departure set. Is like a function. The function has a domain and range. Is something very similar in relations. Relation has a departure set and then destiny set and a graph that models how they are interacting. And this graph is a subset of the Cartesian product between A and B. In this case, we have only one set, the set of general neurons. So A equal to B, and A is included. It's a subset of X. So this A is the set of interacting general neurons. It's like all neurons that are interacting like this. In this case, we have two neurons and astrocyte. This set has the set could be one neuron, another neuron, and the astrocytes. And in simple words, we have this graph. This graph is simply a set of ordered pairs like this. is an ordered pair with one general neuron and another general neuron. This ordered pair stands for a neuron X, it can set neurotransmitter to neuron XG. It's as simple as that. And that set has all the interactions in these synapses that can be a tripartite synapses, a simple synapses, or a gap junction between astrocytes. Well, in this way, we define all of these synapses. As an example, a gap junction. A gap junction is a synapses and that allow the flows from one neuron to another neuron and vice versa. So this graph has both direction represented by these ordered pairs. And that's a bit formal, but we can think in synapses as these sets of ordered pairs, meaning the interactions. But we have more than one synapses between two neurons. As we know, a neuron can be hundreds of synapses between this neuron to another neuron. Some in one way and some others in another way. So we need a framework that cover this complexity. And we have the term in mathematics that index family. What's an index family? All another time and triple. Another triple that has A, that A is the set of all synapses, and I is the set of indexes. So we have that indexes could be 1, 2, 3 until the end, and F is a surjective mapping, a surjective function from I to A. So graphically, more easy to understand, we have this pair that is one synapses. Remember, in this case, one synapses is a simple synapses, one-to-one direct synapses, classical synapses. And we have these indexes. So we have, in this sense, in this direction, from XI to XG, we have 3 synapses. So we have 3 indexes also in the indexes set. So we have XE repeated 3 times, but these are different synapses. And another directions, we have the 4 synapses. In this case, another direction synapses. But we have some conditions. First, the condition that we have at least one tripartite synapses, the minimal condition, because this is a neuroglia network, so we need a tripartite synapses. Another condition is that we can define a sequence from one neuron to any other neurons that belong to a graph in the set of synapses, or the inverted graph. That is that I can I can do this path in this direction if these synapses are in this direction, but I can do that another way, because this inverted graph is like inverting all direction of the network, including the tripartite synapses that is much more complex than simple synapses, but I can invert all these directions with the term of inverted graphs. So, but we can have one neuron that I can reach. There are no synapses, so this is one condition to wholeness. We have one network, not unconnected graph. Then we can see also this complexity as not as an indexed family that is a bit complex. We can simplify it a bit using multi-set of synapses. A multi-set of synapses said, well, I have 3 synapses in this direction and 1 synapses in the other direction, and we have a counting function that says that this direction, this synapse has 3 times, appear 3 times in the set. So and another direction 1 time. This is like a counting function. This concept is quite similar to weighted graph, but it's not weighted graph. It's something more general than weighted graph, because in multiset, each element literally appeared more than 1 time. It's not like a function, only a function. No, it's that the multiset has this element repeated way. many times, n times. So we have this to simplify a bit. It's like simplification because we have this synapse repeated 3 times. It's not like we have 3 synapses, but the same synapses actually. In an indexing function, indexing family, we have these synapses appearing 3 times, but there are different synapses in that mathematical sense. In this case, we have the same synapses appearing 3 times. It's like something like that. If you want to see more about multiset, you can read to Girish. It's a Girish 2009 and all his material. Well, we want to simplify a bit more from multiset to multi-digraph. Probably you know about digraphs. Multi-digraph is just repeating these simple connections many times. If we have multisets like this, we are complicating a bit more. Now we have a gap junction here appearing 3 times, and where a simple synapses appearing 1 time in the multiset. We want to transform this multiset in a multi-digraph. We need to agnes the multiset and only put in some directions that appear 4 times and another direction appear 1 time. And this is a multi-digraph. It has some advantages and disadvantages. The advantage is that multi-digraph as graph has many properties and are very useful in neuroscience, use graph and digraph for network science. And the disadvantage is that we conjecture that it doesn't perceive isomorphisms. In graph, and in general, in category theory, we have a term called isomorphism. Isomorphism, informally, is a structure-preserving mapping between 2 structures. As an example, we have these 2 graphs and are isomorphic. It looks so different, but are the same thing. The same graph in this with different levels. And the same thing can be defined for multi-digraph and epsilon set also. It's just formalizing and generalizing the concept of isomorphism in graph to multi-digraph to multisets. But we conjecture that this transformation does not preserve isomorphism in general. We are working now to know in which case preserve isomorphism, because transforming a multigraph have lots of advantages, and we are working on know in which case it could be work. So I'm not sure if I have more time for the extra results.
Can you wrap it up soon, Marco? We have 2 minutes overall.
Okay, we have 2 minutes. Well, we have
including questions. Sorry, including question 2 minutes.
Oh, okay. Yeah, that's all in this case. Well, and acknowledge to my collaborator, Nelson Castro from Brazil, IMPAM. Thanks.
Thank you, Marco. That was great. Yeah, feel free to ask a question in Q&A. Marco, I think I missed a part that you were talking about astrocytes. What is a specific thing about astrocyte in this modeling?
Yeah, the astrocyte play a role in the network. It's like these astrocytes are connected to single synapses. That's the tripartite synapses. So we want to model this network with these multiple connections. That was the goal of the study.
But you can use it for any neural network, right?
Yeah. In general, you can use this to model with physics or to connectomics, for example. Now we have lots of information about connection between synapses, but we can also reconstruct astrocytes and look their connections and whether the astrocytes are connected with single synapses. Is not too difficult, but we need a framework to model all the network and not only neurons. If you have to model only neurons, you can use graph theories or digraph theory. But if you want to include astrocytes, we need other thing that is not that graph, definitely. Could be an index family, could be a multisets. And we can simplify to a diagram, but we are losing some properties. That's.
Thank you so much. Thank you so much. That was great, Taika, and I am looking forward to your future work. Perfect. Thank you so much. And let me go to the last speaker that we have today. The brain does oscillate, and the frequency has been speculated that they are correlation with the animal behaviors. But it's more than that. Its oscillation are sometimes coupled. We know that using the exciter-inhibitor networks, we can easily find oscillation in the oral network. But how does coupling, especially phase coupling, can emerge from the physiology and anatomy that we see in the brain? Today, Alexandra is going to talk about the modeling of phase amplitude coupling. Alexandra Hatzikamliminiu, I hope that I spell your name right. I'm sorry if it's not. Is a PhD student from University of Toronto working with Dr. Skinner, and she is talking about on the generation of the theta rhythms and theta gamma phase amplitude coupling. Thank you, Alexandra, for joining today, and we are looking forward to hearing your talk.
Okay, I hope you can hear me. Thank you very much, Hervé, and thank you to the entire NeuroMatch grouping for giving me an opportunity to present my work today with an oral presentation. So today I'm going to talk about the generation of theta rhythms and the theta gamma phase amplitude coupling, work that was done during my PhD. So theta rhythms are very prominent oscillatory activities in many brain structures, but they are particularly strong in the hippocampus. This is an area that is associated with spatial navigation, rapid eye movement, sleep, and many cognitive functions. And as a result, theta rhythms have been considered to constitute a very promising biomarker for learning and memory, with applications in pathologies such as Alzheimer's and epilepsy. The C1 hippocampus, particularly, has a large variety of inhibitory cells. However, a cell type-centric explanation of these rhythms is still missing. So my goal has been, in general, to decipher the cellular origin of theta and gamma oscillations and to unravel the biophysical principles of their cross-frequency coupling. The biophysical context, the physiological context that I use for my modeling work is that of an intact in vitro hippocampus preparation that autonomously generates theta rhythms without external inputs from the C3 or the entorhinal cortex. This same area also produces gamma rhythms, which are coupled to theta. This preparation presents a number of benefits. Theta rhythms in vivo are very state dependent as they depend on the particular behavioral state of the animal, and they are very regular. They depend on the specific pathways that are activated at any given time. For those reasons, it's very hard to decipher the cell type contributions to those rhythms and also to understand the fundamental mechanism of theta rhythms. Now, it's also very important to point out that theta rhythms are local field potential activities, and it is very important in computational studies to model them utilizing biophysically realistic LFP models. So LFPs are the summation of transmembrane currents that pass through the cellular membranes at the vicinity of a recording electrode tip. In my work, I've utilized mainly two models. One is a simplified model. I call it the minimal model. It is comprised of simplified cells that they don't have any morphological aspect or biophysical representations for the ion channels and intrinsic properties. Instead, they are Zikevich neurons. They are described by a set of differential equations that reproduces the dynamic output of the cellular activity. I've also utilized the second model. I call it here the detailed C1 model, which is the state-of-the-art biophysically accurate model of the C1 hippocampus that produces these intrahippocampal oscillatory activities. It is comprised of cells with morphological detail, realistic representations of their ion channels and intrinsic properties, and experimentally derived connectivity. Now, this takes me to the first part of my results, in which I'm going to talk about how linking these two models allows us to understand where the frequency of the theta rhythm is generated. The challenge with this biophysically accurate models is that due to their multi-parametric and high-dimensional nature, it is very difficult to analyze them using theoretical tools. So in my work, we propose an approach in which we use hypotheses from the simplified models in application to the biophysically detailed models, and we examine whether those predictions are true. First, we compared the two models to identify commonalities and differences in their structure that could underlie similar or different theta mechanisms. These were two separate models developed by different groups, which, however, represent the same intrahippocampal activities. Then we isolated a piece from the detailed model. We call it the segment model, which is the same in cell numbers as the minimal for meaningful comparisons. We found that the two models varied with respect to the feed-forward excitatory conductances to the pyramidal cell. These are the recurrent pyramidal conductances and the entorhinal cortex and C3 to pyramidal conductances. For those reasons, we focused our investigation around these particular conductances at first. We found that the theta rhythm in the segment model increases with both ECC3 to pyramidal and the pyramidal conductances. The theta frequency is almost anti-correlated, and the stimulation that represents the activation of those conductances increases as the ECC3 to pyramidal conductance decreases. And we can understand how these networks depend on those conductances if we take a look at the inner workings of the cellular activities. Let's take a look at two example cases, a case A and a case B. Case A is characterized by a low ECC3 to pyramidal conductance. For that reason, the stimulation representing the activation of those afferents is high, which means that there is a strong concurrent activation of most of the inhibitory cells in the network. Now, in case B, the ECC3 to pyramidal conductance is large. For that reason, less stimulation is required to induce the same amount of activation of the pyramidal cells, and the activity of the rest of the inhibitory cells is low. This showed us in a very illustrative manner that what seems to be important for the theta rhythms is the input, the net input to the pyramidal cells, regardless of the exact composition of that input, whether it's comprised of strong inhibitory and excitatory currents or weaker only excitatory currents, as in the case B. In fact, those two networks produce theta rhythms of very similar power. So that allowed us to hypothesize that what's really important is this net amount of current that is delivered to the pyramidal cells, and we investigated this hypothesis. So we plotted the theta frequency of any given network as a function of the mean input current to the pyramidal cells, and we found that these two modalities are very strongly correlated. This tells us that the theta rhythm is controlled by that input current, and that input current can predict the frequency of the resulting theta network. We also found that this input current was very noisy, as the network that we analyzed was only a segment of the original full CA1 model, and many of the inhibitory populations hadn't yet gotten the chance to organize into coherently firing populations, which showed us that the rhythm is not imposed by the inhibitory cells, but it is initiated, generated by the very pyramidal cells, so long as the amount of the current is appropriate. Which further suggests that the frequency depends on the intrinsic properties of the pyramidal cells. Now, experimentally, we know that if we remove the PV positive cells, we lose the theta rhythms. We did the same test in our models, and we found that in the case A network, removing the PV cells abolished theta rhythms, but not in the network of case B, which tells us that the contribution of the inhibitory cells is important and more biophysically realistic. As a matter of fact, we found that removing the PV cells increased the standard deviation of the mean input current, which tells us that the mean input current became a lot more irregular and that dysregulated the theta rhythm. Now that takes me to the second part of my results. Can we utilize the understanding that we obtained from the analysis of the segment model in application to the full CA1 model that is comprised of the 8 inhibitory cell types, and it's a much larger network? So we need to notice two particular trends in this network. We have two types of inhibitory classes. First are the OLM and the bistratified cells, which are primarily driven by the pyramidal cells, and for that reason, they fire in a theta-paced manner. And we have the rest of the inhibitory cell classes, which fire in a very noisy fashion because they're primarily driven by noisy external afferents. With that, let's take a look at the last 500 milliseconds of this raster plot. We will first accept the duration of any given theta cycle is controlled by the pyramidal cells and the net input that they receive, and by extension to their intrinsic properties. So let's take a look at a given theta cycle from 3.8 to 3.9 milliseconds. By virtue of the pyramidal connections, more and more pyramidal cells are recruited to the theta cycle, and the numbers maximize at the peak of it. At that point, the pyramidal cells activate the OLM and the bistratified cells. The bistratified cells form very strong connections with the CCK basket cells, and they inhibit them. The CCK basket cells form very strong connections with the PV positive basket cells, and they disinhibit them, mainly activate them. The activation of the PV basket cells induces the termination of the theta burst. So even though the very frequency is controlled by the pyramidal cells, the termination of the pyramidal cell theta burst is controlled by sequential activation of inhibitory classes. So that takes me now to the end of my presentation. In the first part, using the segment model, we showed that the theta rhythms are initiated by the pyramidal cells, and the theta frequency is controlled by the net input, the mean input they receive. And in the second part, showed how the termination of the pyramidal cell theta burst is controlled by sequential activation of inhibitory classes. Thank you very much for your attention.
Thank you so much. That was brilliant. Oh, sorry, if you want to acknowledge people.
Oh, yes. I just wanted to acknowledge my supervisor, Professor Francis Skinner, and my committee meeting members and all my colleagues and collaborators. Thank you.
Thank you. That was a brilliant talk. And I think that there are a bunch of questions are coming up. There are right now 2 questions. Aditi asked that, have you seen any theta frequency change when inputs from only EC, cerebral cortex, or CA3 are considered?
Yes, there is a change in the frequency, but one should consider also the, we'll try to associate that with the mean input current that is delivered at any given time. But definitely when the inputs only from the CA3 are considered, we tend to see a bit of a higher frequency, and CA3 is also associated with gamma rhythms. But I haven't done a proper analysis of that to answer very strictly. But the idea is that probably there will be changes in the frequency even along the septotemporal axis where different kind of inputs are more prevalent than others.
That's great. So I think the second question is also relevant to this one. A broader view, testing the idea that could you assume CA1 network is more suitable for pattern completions in comparison with the pattern separation? Mm-hmm. Is there any circuit-based explanation in results supporting this idea? And the second question that Amin asked is that, could we assume that there is a competitive process at the circuit level between CA1 and CA3, or is there any evidence in your result?
I'm going to stop sharing. I think I can see the questions as well, right? Yeah, so the first question was about pattern completion and pattern separation. So I don't think we can yet speak to those mechanisms in terms of pattern completion and pattern separation for 2 reasons. The first one is that this circuitry explains the intrahippocampal theta rhythm, so not the theta rhythm that we see in vivo, and these mechanisms are in vivo mechanisms, the pattern completion and the pattern separation mechanisms. So the mechanism that I'm suggesting right now speaks to the variability of the CA1 to intrinsically generate these activities, and then how external inputs modulate these rhythms. It's still something that needs to be deciphered. So I would say that we can't deduce a lot just from this mechanism on this question. But I think definitely the mechanism that I'm suggesting speaks very much to the role of the different cell types, which then can be translated in vivo. So I think it's a very important first step. And then the second question is, could we assume that there is a competitive process at circuit between C1 and CA3? Competitive process. Again, to think about this sort of relationships, I would have to think about them in the context of some behavioral state. So I'm not sure Whether I can answer that question generally, I would have to think of some particular behavioral state to sort of think about competitive relationship between the two regions. I mean, the very idea of the main idea of my work has been that the CE1 can generate these rhythms on its own. Definitely, they're a lot weaker than they are in vivo, and the CA3 inputs that I have considered are very noisy, so they are not spike trains, they're not structured inputs. So I haven't explicitly looked at the spike train inputs from the CA3 in this work.
Thank you so much, Alexandra. That was great. And by that, we are on 45 minutes and we have to wrap up these sessions. We had an amazing talk today, and I really enjoyed their talk. Again, thank you all of you for participating today. And I'm pretty sure the attendees also enjoyed the talk. See you again, probably in the rest of the conference.
Thank you.
Hope you have the rest.
Thank you.
So my sense is that the audience are still coming in, so I'm just gonna share my screen and put this up for like 2 minutes while we wait for more people to join. Suddenly got so quiet. All right, Kami, if I understand correctly, we are live right now, but we are not streaming on YouTube, right? We are not yet streaming on YouTube. Okay, cool. Okay, I think it's time we get started. So hello, everyone. My name is Chen Xue. I am a systems neuroscientist from University of Pittsburgh. I study neuronal mechanism for natural decision-making. But as I read through the abstracts for today's talks, it is kind of eye-opening to learn how powerful some of these cutting-edge imaging and video technologies has become, and what they can achieve in, combined with the most advanced analysis techniques. So I think regardless of your background, I'm sure everyone in the audience will feel the way I feel as well during the talks. So the 3 talks today are truly for everyone in every neuroscience field. So please feel free to ask any question that interests you, even if you feel it's a dumb question. My whole job today is to be the dumb guy for you. That's the kind of exclusive service you get only during a pandemic, I guess. So without further ado, let's hear from our first speaker, Fazaneh Najafi from the Allen Institute. She is going to tell us how cortical connectivity across columns changes based on past experiences. So Fazaneh, the floor is yours. I will stop my share so that you can come on stage.
Hello, everyone. Can you hear me?
Perfect.
Yeah, awesome. Okay, so let me start talking. Thank you, John, for introducing me. As he mentioned, I'm going to talk about a novel cutting-edge microscopy technique, which we have named multiplane mesoscope. Then we used it to ask some important questions in neuroscience regarding the representation of expectation violations. First, the scientific question that we are interested in. It's about predictive processing framework. This framework assumes that the brain contains an internal representation of the world. And it generates predictions about what's going to happen about the external world, and then it compares them with the actual sensory inputs, and then it keeps updating the predictions. One thing about this framework is that it relies on communication across different brain areas in order to generate and update the predictions. And of course, a major challenge is to simultaneously measure neuronal activity across multiple cortical areas. So to address this challenge at the Allen Institute, we have developed multiplane mesoscopes that allow simultaneous imaging from 8 cortical planes. The original mesoscope was developed in the lab of Karl Svoboda in Genelia. At the Allen Institute, my collaborators adopted that scope and doubled the throughput of it by mainly adding 2 major features. One is temporal multiplexing, which is about splitting the laser beam into 2, delaying one of them, sending each of them to an remote focusing unit. So in addition to temporal multiplexing, they added an additional remote focusing unit. And then these 2 beams that are split and delayed from each RFU, they end up being focused onto 2 cortical planes. And then the signal from them is collected and demultiplexed, and that's how we get to image 2 planes simultaneously. Then through galvo mirrors, it's scanned, and so we get 4 sets of these plane pairs. And that's how we get to image 8 cortical planes simultaneously. One of the modes is to do 2 areas and 4 depth, which is the one that I'm going to be talking about, but also the scope can be used in an alternative mode, which is 2 depth and 4 cortical areas. So the data set that I'm going to present involves 2 visual areas, V1, the primary visual cortex, and LM, which is one of the higher visual areas in mouse. 4 cortical depth, roughly corresponding to superficial layer 1, 2, 3, 4, and perhaps superficial 5. And then the data is collected in 3 mouse lines that are tagged for excitatory neurons and 2 inhibitory subpopulations, SST and VIP. These 2 inhibitory subpopulations are especially of interest. Why? Because VIP in particular receives inputs that are related to arousal, animals' locomotion, and also associational inputs from other cortical areas. It mostly synapses onto SST neurons, and as a result, it has a disinhibitory effect. So we were interested to look at the activity of VIP, SST, and excitatory neurons during a visual task which involved violations of the animal's expectation. And what do I mean by that? So the animal is on a disk, and then a series of images are shown to the animal. Each image is 250 millisecond, followed by 500 millisecond gray. And so the animal keeps seeing these images, but sometimes unexpected to the animal, they're omitted. So this is one of the unexpected events that happen in our task. There is another one which I will talk about later. So the expected events are these images because they come repeatedly, and the unexpected one is the random infrequent omission of the image. So let's look at the activity of each cell type in each visual area and in each of the cortical depths. First, let's focus on V1, excitatory neurons. What you see here, these boxes are when the images occur. This one shown in dashed is the omission. Red, let's focus on the red. Red is a superficial layer 5 or deep layer 4. So let's see what layer 4 or 5 in V1 does. When the image comes, the activity robustly goes up. And then after the omission, we don't see much. Then the activity again goes up for the next image and so on. This is expected from excitatory neurons in layer 4-5. If we look at the more superficial layers, we see a similar response, but very nicely it's graded. So we see a more or less similar pattern also in the LM as a higher order visual area. So in excitatory neurons, we see this robust and graded representation of images according to the cortical depth. When we look at SST, somatostatin inhibitory neurons, we see a similar pattern, a very robust activity driven by the images. And also, interestingly, there seems to be a little bit of inhibition going on here. So in SST also, we see these cell types very much like images, and their activity seems to be a little bit inhibited. We don't quite see that graded representation across cortical layers. In this layer 2-3, we see more of a response, but the rest of them are not that different. So SST and excitatory neurons are more or less similar. What's very interesting is when we look at VIP neurons. For these neurons, first of all, notice that they are not really recruited by images. If anything, there is a suppression here. And instead, there is a small pre-stimulus ramping that precedes the image, as if they are predicting the image occurrence. And then what happens after an omission is that this ramp keeps going up. So VIP neurons respond very differently. They ramp prior to the image, and they represent omissions very robustly. An earlier paper at the Institute has represented some of these results as well. So we think VIP neurons might have something to do related to providing predictions and perhaps prediction errors for the cortex. But that's a hypothesis and needs further testing. So these are just population averages. And of course, we thought we might have missed something. So next, I did use a population decoding. I used support vector machines to study how the response of these cell types changes after images and omissions. The results after images were just how the population averages had suggested. And also I have subsampled neurons, so we can do a fair comparison across cortical depth. So we see a strong representation in our deepest plane, and then smaller in more superficial depth. What is very interesting is that after the omissions, the population decoding revealed a strong signal in the excitatory neurons. That's in contrast to what we saw here, which on average, excitatory neurons were not showing any response after omissions. So what this is suggesting is that even though on average we don't see anything, but at the population level, excitatory neurons carry signal about omission. And so there must be some small percentage of excitatory neurons that are representing omissions. And so with a optimal decoding methods such as SVM, we are able to capture that signal. In SST as well, not only we see that strong response population signal after images, but also now again, we see that omissions are also represented by SST neurons. For VIP, the picture was not that different from what we got from the population averages. So in brief, both our average and SVM analysis reveal that excitatory and SST neurons carry a strong population signal after images, which are the expected events, also after omissions. VIP neurons show a small ramping before the images and a very strong representation of the unexpected event. So remember, the key feature of our scope is that it's simultaneously imaging these 8 cortical planes. So of course, we wanted to see how the different planes and the 2 columns interact with each other. So here I did a correlation analysis. I basically looked at pairs of neurons and how their activities co-vary after image presentations and also after omission presentations. Let me walk you through these figures. This is for excitatory neurons. Here is the LM and the depth of LM. We're going from superficial to deep. For V1 also, we're going from superficial to deep. And this is the value of the correlation coefficient. The brighter means the higher the correlation coefficient. So what we're seeing here is that after images are presented, there is a strong correlation between the deepest plane in LM and the deepest plane in V1. This is interesting and in line with the literature that when there is this thalamic input going to V1 and perhaps LM as well, and that's why we see this correlation increase in the excitatory network after images in these deep layers. After omissions, there is not much
Sorry,
really.
Padane, we would
Sure.
appreciate if you wrap up in 2 or 3 sentences.
Okay, sure. For the SST neurons, we also see a similar thing, except it's not quite in deeper layers. And the main finding is that in the VIP network, we see that after omissions, there is correlation, and it's mostly in superficial neurons. So the gist is that the excitatory neurons and SST neurons become more correlated after images and in deeper layers. However, VIP neurons after unexpected events become correlated and in superficial layers. On Thursday, my intern, Akul, will be presenting a recurrent neural network analysis of the same data set. The main finding there is that when we change this set of images that the mouse was looking at, so basically the statistics of the environment changes, we see that there is a change in the feed-forward and feedback connections. Because remember, with correlations, we cannot get directionality. All we got was that correlations go up between these planes or down. But with recurrent neural net training, now we got to see how V1 to LM and LM to V1 connections are different after familiar images versus after novel images. And very briefly speaking, you can go to his talk, it's on Thursday, but he'll show you that after novel images, we see that the feed-forward connections increase in deeper layers. However, the feedback connections are stronger in superficial layers. The conclusions are that multiplane mesoscopes allow us to image at different cortical depth and areas simultaneously. With this instrument, we demonstrated how interaction across cortical columns are different after expected and unexpected events. Unexpected events recruit activity primarily in the VIP network and in superficial layers. Expected events recruit activity in excitatory neurons and SST neurons. RNNs revealed that feed-forward and feedback connections undergo significant changes when the environment statistics changes. The multiplane mesoscope data will be also released by the Allen next year. That will be available to the public. You can download it and use it for your data analysis. Finally, conclusions. My manager, Jerome Lecoq, both on the scientific side, he was a great person to talk to, and also he was a key member of this group. led by Peter and Natalia and Dimitri, who developed the mesoscope, and a group of RAs, Sam, Sarah, and India, who collected imaging data, and my colleagues at the Institute, Marina, Doug, Sean, and Peter, who were key members of the behavioral pipeline. Also,
Right.
thanks
Thank
to
you,
all
Farzaneh.
Alice and Neuromanagement organizers. Sure, yeah.
All right. Thank you. I'm clapping on behalf of the audience here. I have put it down in the notes for at least 3 questions, but unfortunately we don't have time for that. So thank you again. If there are any audience questions coming up, Farzaneh, you can still answer the audience questions in the chat window, right?
Sure, I will.
So, okay. So our next speaker, Thomas Athay from Johns Hopkins University, that I have to say that whose work involves the most mesmerizing pictures I see in neuroscience. So Kami will post a link related to his work, and I encourage everyone in the audience to check it out. So Thomas, ready to show us some pretty pictures?
Yeah, absolutely. Thank you for the introduction, Chang. Can I just get a thumbs up that you can hear and see my slides? Awesome. Okay, so I'm going to talk about some work in progress about automated neuron tracing. And just to start with some background, so whenever you talk about neuron morphology, you kind of have to give a shout out to Ramon y Cajal, who was the first to kind of sit down and in a diligent way look at different neurons and categorize them according to their shapes, according to their morphologies. On the left is one of his beautiful drawings exhibiting different neurons and their different morphologies. But neuron morphology is not just a thing of the past. In contemporary research into projection neurons, morphology plays a key role. For example, in defining the major subtypes of projection neurons, we have neurons that go from cortex to cortex, those that go from cortex to thalamus, and those that project below the cerebrum. But morphology is not just relevant in subtyping and classification. It's pretty key in understanding learning. So in a classical neuroscience experiment, the figure on the left here shows that in an experiment where they sewed shut the eyes of some kittens and then studied the visual pathway between eyes that were sewn shut and eyes that were allowed to be open and look at the visual world, there was a difference in the morphology of the axonal arbors here on the bottom and on the top. But not just learning, in evolution, so us as primates, humans, we have enlarged upper layers of our cerebral cortex, and those are associated with particular morphological subtypes of projection neurons, in particular the ones that project to other locations in the cortex. And if I haven't motivated you enough, I think we're all motivated by research funding and the Brain Initiative Cell Census Network, which is funding a lot of neuroscience research in this area, has the explicit goal to identify different brain cell types, and morphology is one of the key pillars in that work. Oops. Okay, so neuron reconstruction, or the tracing of neurons in order to study morphology, is not a new research area. There's been a lot of research into it, in particular in the past decade, for example, under Diadem and Big Neuron, these initiatives where many different solutions were proposed, deep learning solutions, reinforcement learning solutions, and a host of others. However, the primary limitations of the existing methods are that they assume there's a single neuron in the image. The methods don't really extend to projection neurons, and some of the solutions, in particular the deep learning ones, rely on a lot of voxel-wise ground truth, which is hard to come by. So the data we're working with comes from the Janelia Research Campus, in particular their mouse light team. So we have some complete mouse brain images from 2-photon tomography that are at the submicron resolution. And you can see these colored lines here are reconstructions of projection neurons. And this process is already done in a semi-automated way. However, it's still the bottleneck in producing these data sets. So my problem, or my research is into making this more efficient. So I'm going to kind of describe the algorithm I'm working on. Any good imaging pipeline starts with some pre-processing. Basically, the important thing here is that we can apply a neural network to predict whether a voxel in the image is part of foreground or background, part of a neuron or not part of a neuron. And in most computer vision tasks, this would be all you need to do. Is this voxel part of the sky? Is it part of the road? Is it part of a person? And then you would be done. However, the shape of neurons is these kind of spindly things with long long axons and these curly dendrites means that if you have a false negative anywhere along the axon, you could cut the neuron completely in half. So the existence of these false negatives means you're going to have a lot of fragments that you need to string together. So on this animation is just kind of illustrating some of the different neuronal fragments that may be predicted by a neural network. And this is just a few of many. And the problem now becomes, how do we string these together into a complete neuron? And for that problem, we turn to hidden Markov modeling. So hidden Markov modeling is a classic statistical modeling technique where we have two sequences of random variables, one of which, the one I denoted with X, is unknown or hidden. And the other one is observed. In this picture, it's Z. And the classic use of this hidden Markov modeling was back in the 80s with speech processing, where you observe this audio signal, which would be Z, and you're trying to estimate the words that gave rise to that audio signal. In our case, what we're trying to estimate is a sequence of image locations, kind of a path winding through the image. And what we observe is parts of the image associated with that path. So the real beautiful thing about hidden Markov modeling, the reason why people do it is because estimation is very efficient. So the problem of computing the maximum a posteriori estimate of that sequence of hidden variables, or the map estimate, becomes just a shortest path problem through a graph. So I'm trying to estimate the most likely hidden path according to the posterior distribution, and this becomes a shortest path problem. So in the little animation here, I have the classic illustration of finding the most likely sequence of states from left to right. So the different rows are the different possible states, and the different columns are the different elements in a sequence. And you can estimate the most likely path with what's known as the Viterbi algorithm. It's an example of dynamic programming, and it works in an iterative manner as shown in the animation there. So I want to give a brief video of the Viterbi algorithm working on neurons. It's kind of a short video, but before I get it started, I just want to explain that the green sections, so you'll see the green, and that is the most likely neuron path at that given iteration. And then the red paths are kind of the less likely but still attractive alternative candidates. And you'll see the green is kind of moving along, and the red is out exploring until the green makes it back to the soma. So you can see the red is out exploring over here. It's kind of going to the cell body. It's going to other dendrites here, and the green is hanging out until it comes all the way back to the soma. So this algorithm would work. You would kind of place a bunch of points distally, and then trace all the paths back to the appropriate cell body. And it's worth noting that this path could have gone to these other cell bodies, but those paths were not as likely in terms of posterior probability, and thus did not arise in the segmentation. So this is just a static image of kind of you would have points be placed distally on the neuron, and then it would trace them automatically back to the soma here. So we have a grayscale image. I have red highlighting kind of the segmentation or the trace that is being done from each of these green starting points. So what I'm working on now, I definitely have a lot of work cut out for me for the rest of my PhD. The main focus is scaling this up. So I kind of showed you a 200 micron approximately cubed subvolume. I need to scale it up to the whole mouse brain, which will really be leveraging the dynamic programming to do that. And then we'll also need to quantify the performance of these reconstruction of this reconstruction algorithm, and there are established ways of doing that. So that will be straightforward. So thank you all for coming to join me in this talk, and thank you to all the people on the screen for helping me with the material that I presented. So I'm glad to take any questions at this point.
Thank you, Thomas, and thank you for keeping on time. And those captions were great. And my sense is that the audience is still taking time, typing questions and voting for them. So I'll just ask the straightforward ones. I'm wondering how much you have to tune your model to fit a specific set of data. There are other data. There are other data sets out there. For example, I know Alfonso Silva, is that probably the name, who worked on marmosets, and where I think they took a heroic effort of doing a lot of these things manually. So I'm wondering if you apply your models to data like outside of the ones you presented, how well do you think it will extrapolate?
Yeah, so that's a good question. I think the key, so my two comments with regards to that are, one, probably the most, so I acknowledge this mouse-like data set is very high quality. I think it's a big reason why, where I get to show cool animations. So it's definitely true. If the images are not good, there's really nothing, there's not much that you can fix on the computational side. You really need to start with good images. I think the main key of why I would see it generalizing or not generalizing would be kind of how the level of sparsity of labeling in these neurons. So there's about 100 or so neurons per brain in the data sets we're working with. And I don't, in working with the different brains I have been, as long as that level of sparsity is around the same, it's pretty easy to deal with things like, mean shifts and stuff like that. However, I could see it failing if a ton of neurons in the same spot are all labeled. The final comment I want to make is the longer term plan is to make this kind of an interactive thing. Because the algorithm works in an iterative manner, we can flag the points at which the algorithm is unsure where to proceed, and then like a manual reviewer could come in and efficiently figure out what the appropriate path is. So I think for those reasons, it should generalize to images of similar sparsity and of similar quality.
Thank you, Thomas. I think that pretty much covered the question from Benjamin Pedigo, but I'm going to ask it anyways in case you have anything to add. So he said, would the dynamic programming approach be fast enough that it could work in real time with a human annotator, as in, help improve the semi-automated approach? And I guess the answer is yes.
Yeah, certainly. For one neuron at a time, which would be as much as a human could handle. It's as much as a human does in the real world anyway. Yes, it could.
All right, thank you. And we need to move on to our final speaker of this session, Christian Ebersen from NYU. He is dealing with a completely different beast, which is behavioral tracking of freely moving animals. So Christian, I'm excited to see how you get a hold of these scurrying little mammals.
Okay, can you hear me now? Okay, good. Yeah, thanks so much. So I will talk about a method we've been working on for tracking socially interacting mice. So I'm a postdoc in the lab of Robert Fromke at NYU. So motivating a little bit what we do, a lot of kind of social behavior studies that we neuroscientists do in the lab are something a little bit along the lines of this. But actually, social behavior is very complicated. It's very three-dimensional. The animals kind of move in complex ways over each other, around each other. So would be nice if we could track these animals and track their postures in more than just, or in full 3 dimensions. So I'll go briefly over. People have tried measuring animals in 3 dimensions in the last few years. Here there's a paper where they used a kind of motion tracking system, kind of similar to what you do in movies for a single rat. Other people have also used depth cameras to track mice. Some approaches are built on tracking key points in single 2D views and then kind of using triangulation to put them together. These are quite nice, but very sensitive to occlusions. So they're not ideally suited for multiple animals. Another idea people have used is to kind of film using depth cameras in full 3D the whole scene and kind of make a physical model of the animal and kind of reconstruct. Here, for example, there's a paper with 2 interacting rats, or there's a paper with a monkey, and they kind of build a physical model of the whole monkey to track how the monkey is moving. Yeah, great idea. So that's what we wanted to try to do. So I've built a setup that uses 4 of these depth cameras around a kind of transparent enclosure for the mice. These depth cameras are from Intel. So they're pretty cheap, but they're quite nice actually. And they have an infrared laser in the middle. They have 2 infrared cameras, and they also have a small color camera. So what we can film with these cameras, it's for instance, infrared images where you have these dots from the laser. You can calculate the depth image using the 2 kind of infrared eyes, and you can align the color image to these depth images to kind of estimate what the color of pixels are in space. So the idea of an experiment is like this. Yeah, we record from the brain of one mouse, socially interacting mouse, record the behavior with these cameras that record color images, depth images, and they also record the blinking of an LED that's visible for all the cameras. So we can synchronize everything together, synchronize it to the neural data. And this LED is very precise. It's very stable. It drifts a little bit, but you can actually correct everything together and get a very stable system. Okay, so I'll tell you a little bit, just kind of give the rough idea of how we process this data. So one thing, it's these depth images. The other thing is the color images. So for the depth, or actually, what do I say first? Yeah, for the depth, we need to put all these cameras into the same frame of reference. So the way I do this, it's very kind of low tech. I just film a blue ping pong ball that I move around and film with all the cameras. And then we use a few kind of computer vision tricks to find in the color space and extract. Can you actually see my mouse?
Yes, we can.
Okay. So we extract the pixels corresponding to that ping pong ball. And then every camera will have a view of where that ping pong ball is in space. And then we use a kind of robust way to put it all together in one coordinate system. And it's actually quite precise. So a few millimeters variance on the estimate of the center of a ping pong ball moving around this arena. It's pretty good. Then we use a few other tricks, for instance, to automatically find where the floor is, put it nicely into a kind of XY plane. We find the imaging artifacts associated with this behavioral arena, remove those, and then we end up with data like this. So this is a 3-dimensional point cloud. It's kind of the surface in 3 dimensions of the 2 mice here, and one has a neural implant on the head. And I've colored here in different colors the pixels that come from the 4 different cameras. So we can see we put it all together. yeah, so here's an example. This is my hand and some ping pong balls in full glorious 3 dimensions. And it's a full 3D video, so things are moving around. For example, here's a mouse in 3D. And yeah, it's a video. So the mouse is kind of moving around. For the color video, we train a deep neural network to recognize a few key points on the mouse, like the tail, the nose, the ears. And for this guy with an implant, we also recognize where the implant is. I won't go into details, but we use a few tricks to make this work. One of the tricks that works very well is this training the network to recognize also these affinity fields. So instead of only teaching the network to find these key points, we also force the network to learn a kind of more holistic idea of what a mouse looks like. So the network is also learning what is it supposed to look like between a nose and an ear, and incorporate that into its kind of estimate of whether something is really a nose or not. So just to give an idea of the pipeline, we get color video of the mice, depth videos of the mice. Then we have a deep neural network find these key points. We put those into 3 dimensions, and then we can assemble it all together. So now here in the full preprocessed data, the black dots are this kind of 3-dimensional surface, and the colored dots are these key points that we know what are. So how do we find the mice? Well, a mouse, it's kind of like roughly approximated by 2 ellipsoids that are a little bit stretchy, that can move a little bit like hinges. And then we write, okay, except one has this big implant on it and a cable introducing artifacts, which we just model as a ball. And then we write a loss function to find where the mice are in every of these 3D frames. So one term in this loss function is associated with these black dots, which is kind of this 3-dimensional surface. So there's a loss term to the surface of this mouse model. And then from all the colored points, there's also a loss associated with where they should roughly be on the body. Like the nose should roughly be at the end of this ellipsoid, or the tail should roughly be at that end of that ellipsoid. And then if you do a few approximations, you can write it all as pure linear algebra, which is nice because that can be very fast, and you can run it on a GPU. So it's all tensors going into tensors, going into other tensors. And it works. So here I'm showing you kind of a nasty frame. Lots of artifacts from the cable, but you can kind of see there are 2 mice, and you see there are some ears, there's a tail, there's an implant here, there are some ears, and there's a tail. And we use then a particle filter to find in every frame where the mice are. So here I'm showing you how the particle filter for a single frame converges. So robustly, it will kind of converge down to estimate. Here there's one mouse, here there's another mouse that has an implant. Oh, this is what this triangle is. And we do that for every frame. So for every frame, we get this kind of rough, noisy, but very robust estimate of where the mice are in 3 dimensions. But okay, it sucks because it's very noisy. So we also do some filtering, Bayesian filtering tricks, because the frame rate is quite high of these images. They can be up to 90 frames per second. So even though every estimate is quite noisy, if you look here on the right, integrating across frames in a kind of clever way, we get actually a very nice, smooth estimate, which looks pretty good. So you see here, the 2 mice are moving around, even this one guy that has a big implant on the head, even with all the artifacts, it kind of robustly tracks where the 2 mice are. So here on the right. Okay. So what can we do with this data? One thing we can do, I'll just give kind of headlines. It's you can, for example, train also a kind of Markov model to automatically recognize different movement patterns. For example, you can train it to recognize when the mice are rearing up and down, or when they're turning left or turning right, or standing still or moving forward. So you can automatically label all your movement patterns into different states. You can recognize automatically social events, like here on the left, every time they put their nose together. This is a kind of social facial touch. Or actually, let's watch this again. Look here in the middle, when one is sniffing the kind of genital area of the other guy. Yeah, we can recognize that automatically. Or you can recognize kind of postural things like the 3D head direction here, not only of the guy that has the implant, but also the partner. using where the ears are, you can estimate, even when they're quite close, socially interacting, you get a pretty good estimate of what the three-dimensional heading direction is of that animal. And another thing important for the social life of mice, it's that they are actually kind of afraid of light. So we can also do it using only the infrared data. So here, the infrared spectrum, we can recognize, or we can record a kind of black and white image. But the problem is, it's not just black and white. It also has this laser artifact picture, these laser dots everywhere. But there are ways to train a neural network to disregard that kind of noise. Like, for example, if you are driving a self-driving car and it snows. Yeah, you don't want to die. You want the network to still recognize what's going on. So we train the network to disregard these kind of bright spots. And here I'm showing you then a few mice recorded in darkness. So in only the infrared, under infrared light. So it's much more comfortable for the mice. Their social behavior is kind of much more natural. Yeah, and then the code is not totally on GitHub yet, but soon it will be on GitHub. And people are very, welcome to contact me if you're interested in using this for an experiment. And then, of course, I want to thank Rob, my PI, and the whole lab. And there is a preprint about this already on bioRxiv. So if you're interested, you can also find it on bioRxiv or send me an email or something. Yeah. Thanks so much. Are there any questions?
Thank you, Christian. That was super cool, I have to say. So you kind of showed this, right? So you identified, you have two mice in a jar, and you can identify one from the other, but one has an implant. How well does it work for like mice without implant? Are you able to build individual models for each animal?
Yeah. So actually, it works also quite well when they don't have an implant. It's a much easier thing actually to do, I would say. And now there are actually also other tracking methods that can track mice if they don't have implants and don't have cables and all those artifacts. When I started making this, there was no multi-animal deep lab cut or no multi-animal leap or anything like that. But now there are also other methods that you can use. The 3D stuff helps you a lot. A big problem normally, if you only have one view and the two mice get close and then one passes over the other one, you lose track of who is who. But if you track them in 3D, you can kind of see, who is going over who's going under, and who's coming out on the other side. So in that sense, the three-dimensional information helps keeping track who is who. So you don't do it by how they look. It's purely by the kind of spatiotemporal continuity.
I see. So it's going to be a little bit more challenging when you have more environmental enrichment in the cage or whatever, right?
Yeah. That's the next thing. It's to try having, you can also have objects that they interact with. Like you can have a deformable, movable objects would be fun to try letting them play with things, see how they do that.
Okay, cool. We have 1 or 2 more minutes. If the panelists have any questions, you can simply unmute yourself and ask. You don't have to type or anything, since you're already on stage.
I have a question for Fasane about your talk. So the mice are running, and then you emit one image. This decoding you showed with the support vector machine, that's decoding that particular image that's missing, or do they know what image is coming?
Well, actually, that's a great question. What I showed you there, I am not decoding specific images. What I'm decoding there is each time point relative to the gray screen. So as long as the signal is different on that time point compared to the gray screen or the background activity, you'll see something. So if the signal after omission is different, if the population activity after omission is different from the population activity during the gray screen, you'll see something. But now I am doing like image specific decoding, which I did not show. That data is not shown.
So there is a sequence that the mice can expect,
exactly.
What image is gonna
exactly.
come. Okay.
Yes. Yeah.
Interesting. I mean, in the human stuff, there's all these big ideas about predictive coding or mismatch negativity, oddballs, all that. So you think that's all the VIP neurons?
So usually, actually, in that literature, they mostly think it's SSD. They think it's not PV. They think VIP and perhaps neuromodulatory inputs might have a role. SST is shown. There is work that they manipulate SST and mismatch negativity goes. For us, it's interesting that SST, yes, with the population decoding, I saw the signal. When we look at individual neurons, a few percentage are representing, but it's VIP that is showing this massive omission. So one thing is that this is not quite a deviant, right? So it's not like a different image. It's an omission of an image. So maybe with this particular type of error, VIP is mostly recruited. The other thing is that we have this periodic sequence that is also different. A lot of those mismatch negativities, although yes, some of them also have the periodic representation, but a lot of the predictive coding is not necessarily like periodic. It could be just one CS predicting a US. The other thing is that some of the changes might happen at the level of the dendrites, which I have not analyzed. This is all somatic signal. But yeah, we see much less in SST and a lot more in VIP, which is a bit different from the common predictive coding literature.
And where the VIP?
I think we have to, if you want, you can keep the conversation.
Message me, Chris.
Yeah.
Yes, it was really a nice time discussing with you guys, and I appreciate your talks, and I also appreciate the audience for the attention and the questions. And the next bunch of sessions starts in 15 minutes, I think. So you will have time for a cup of coffee or a break or whatever. Thank you very much and goodbye.
Thanks so much.